Hello dear students. Today we are going to revise four chapters which are most important for your NEET exam, right? We'll rapidly revise these chapters: biological classification, plant kingdom, morphology and anatomy of flowering plants. Okay? I hope that you have already prepared very well, right? Whatever important points are left, we'll start reading them along with solving many questions, right? So today first chapter is biological classification. Now in this chapter, you must remember that this chapter begins by talking about the classification of living organisms which are present on the planet, right? So starting from the beginning, many scientists classify different type of living organisms, but the very first classifications were actually non-scientific, right? All the living organisms which are present on the planet, they all are actually classified easily on the basis of the use they provided to us, right? Or you can say on the basis of utility, like any particular organism is good for providing shelter, so they have categorized as of that category that these are shelter providing organisms, right? Like certain trees and all. Many organisms we were using for clothing, right? So they have categorized it in that category that these are good for clothing, right? Or maybe many organisms are good as they can provide us food, so they will be classified as food providing organisms, like, right? So these are certain organisms which were classified on the basis of utility they provided to us. This was absolutely a non-scientific type of classification, right? Then scientific advancement began, and we had the very first type of scientific classification which was given by Aristotle, right? So maybe you have studied about Aristotle already. Aristotle is the person who classified all the plants and animal based on simple morphological characters, right? So you just imagine like this, that at that time when Aristotle started dividing different living organisms, at that time science was not this much advanced, right? And as a result, we were able to classify the living organisms only on the basis of the external characters which are visible to us, right? So on the basis of those external characters, on the basis of the morphological characters, Aristotle divided all the animals under two categories. Those animals which were actually having red blood cell, they were considered as enema, and those organisms which were not having red blood cells, they were categorized under the category of anema. Before any biological word, if it is added, it means it is absent, right? So red blood was present, red blood was absent, just two broad categories. Now you tell me one thing, how can it be a morphological character, you must be thinking, right? So you have to remember, he just observed at the color of blood, right? There are certain organisms you will see that their color of blood is red, right? But in lower group of organisms like insects and all, right, there is no red color in the blood. So he categorized this also on the basis of the color of blood, right? Whether it is having red color or it is having no red color. And you know that red color appears because of the red blood cells which are present in the blood. Okay, then he classified all the plants under three categories. Plants which are green in appearance, he called it as herbs, right? Green in appearance means they will be green only, they will not become woody, they will not become brown, they will not turn into brown plant, right? So all those plants which remain green, which are small, which are herbaceous, he considered them as herbs. All those plants which are bushy, right, but they are not having a perfect thick trunk, right? They are very bushy, they are woody but bushy, right? He considered them as shrubs. And all such plants which were having a thick trunk, woody trunk, he considered them as trees. So this is how he was able to classify the living organisms based on very basic type of scientific character, that is only external character or morphological characters visible to us, right? Then obviously this classification does not hold true. You know very well that we cannot classify all the plants under these categories only and all the animals under these two categories only. So what we have to do, this classification system was revised, right? And one more type of classification system came, that was two kingdom classification. This two kingdom classification was given by Carolus Linnaeus. About him you have already studied in the first chapter, Living World, right? He is father of taxonomy. He is the first, he is the person who actually classified all the living organism based on presence or absence of cell wall. His basis for classification was cell wall is present or cell wall is absent. This is very important to remember. Many times they will ask us questions on this, right? He categorized all the uh, organisms under two kingdoms, right? Two kingdoms are Plantae and Animalia. Okay, kingdom Plantae and Animalia. Now you must remember, all such organisms which were having this cell wall, they were all classified as plants, and all those you can say organisms which lack a cell wall, they were all classified as animals, right? So you just think about all the living organisms starting from kingdom Monera to last, right? How many organisms in this, they do have cell wall? So you'll be knowing that all the bacteria have cell wall, right? Except mycoplasma, right? Protista, starting from chrysophytes, diatoms, right? Then euglenoids, slime molds, sorry, euglenoids only, not slime molds, okay? Euglenoids, up to euglenoids you have, sorry, only two of protist, right? That is chrysophyte and you can say, I'm writing here, right? If you consider all the protists, only few have cell wall, right? And in that also you have to remember chrysophytes, that includes diatom, dinoflagellate, right? Okay. So all the protists which include chrysophyte and dinoflagellates. In chrysophyte you have desmid and diatom. Dinoflagellate, you remember goniolax is there. So these organisms which belong to kingdom Protista, they were categorized under Plantae. But euglenoids, right, slime molds and protozoans, these three from protist, they does not have cell wall, right? They have pellicle. The euglenoids have pellicle, right? So they were classified under kingdom Animalia. So some members of protist were here, some member of protist were here. Fungi has cell wall, right? So it should be considered as plants, right? And all the plants starting from algae to angiosperm, algae, bryophyte, pteridophyte, gymnosperm, angiosperm, all were having cell wall, right? Sometimes they give a question, they will ask you a particular member from this kingdom, and they want to know that this particular member will be a part of kingdom Plantae or Animalia as per two kingdom classification. So you have to remember this. In Animalia, all the animals and mycoplasma along with these members from Protista were kept, right? Now, two kingdom classification you understood. Now, after this, there were so many classifications like three kingdom classification, four kingdom classification, right? Which lead to finally the classification which we follow right now, that is five kingdom classification, right? So, two and three kingdom, sorry, two kingdom classification and five kingdom classification is given in NCRT. Three and four kingdom classification is not given in NCRT. So what is the three kingdom classification? This was given by Ernst Haeckel. See here you have only kingdom Plantae and kingdom Animalia. These two kingdoms are there, right? In three kingdom classification, one more kingdom was added, that was kingdom Protista. This kingdom added for the first time here. Please remember, I have written here that these members of this kingdom, as we know today, they should be a part of kingdom Plantae. Members of this kingdom should be a part of kingdom Animalia. It does not mean that this kingdom was introduced in Linnaeus classification system. Okay, in first page of NCRT it is written that two kingdom classification was that particular classification where all the organisms were divided under two kingdoms on the basis of presence or absence of cell wall. Because you know today all organisms like five kingdom classifications, that's why I have written it like this, right? So you have to be very careful. Kingdom Protista, which was the third kingdom after Plantae and Animalia, was introduced by Ernst Haeckel in his three kingdom classification, and here the meaning of Protista was all the unicellular organisms must be kept here. Whether they are prokaryotic or eukaryotic, that was another case, but all organisms which are eukaryote or prokaryote, both which have unicellular condition, they must be a part of kingdom Protista, right? Then a problem will appear that prokaryotes will come with eukaryotes, right? So Copeland introduced fifth kingdom, that was kingdom Monera, right? And here the meaning of Protista change. Here Monera means prokaryotic unicellular organisms, right? And here Protista means, okay, eukaryotic and unicellular organisms. Are you able to understand? Meaning of Protista is not same throughout kingdom, three kingdom and four kingdom classification, right? So NCRT has written a line that all this classification system may, these two kingdoms are always fixed because they are coming from beginning from two kingdom classification, right? Plantae and Animalia are constant kingdoms, and the members from these two kingdoms are shuffled into other kingdoms in the next successive classifications. Understood? This line is given in the first page of this biological classification chapter. So you have to remember what is the meaning. Meaning of kingdom Protista in five kingdom classification and four kingdom classification is unicellular eukaryotes, right? Meaning of kingdom Monera in five kingdom classification and four kingdom classification is unicellular prokaryotes, right? And meaning of Protista in three kingdom classification is only unicellular organisms. So it is very extremely important to realize the importance of that particular point given in NCRT, that meanings of these kingdoms have been changed as we move through the successive classifications, right? Only these two kingdoms are fixed. Plantae means those organisms which have cell wall, which are green in color, right? Here Animalia may, we have to remember all those organisms which are heterotrophic, right? Which lack cell wall, right? All the members initially are present here. Then as we introduce new kingdoms, the members from here are transferred to different kingdoms in uh, different next classification systems. Okay, I hope you have understood it. Now today I told you that after this three and four kingdom classification came the five kingdom classification, which we are actually following. This is R.H. Whitaker's classification system, which he has given in 1969, and he classified all the living organisms under five kingdoms, right? And he classified these kingdoms on the basis of what all characters. This was a P by Q. Many times they have asked this, right? So he classified all the living organisms under these five kingdoms based on these characters. What is the cell structure? Whether the cell is prokaryote or it is eukaryote, right? What is the thallus organization? Which kind of thallus it is? Whether the organisms are made up of single cell or they have multicellularity also. They have only cell level organization, tissue level organization, organ level organization, organ system level organization, right? All these things are given in one table if you remember, right? What is the mode of nutrition, right? The organisms are following heterotrophic mode of nutrition, autotrophic mode of nutrition. Within autotrophic mode of nutrition you have different categories. Within heterotrophic nutrition you have different categories, right? Reproduction, what is the reproduction, right? Whether it is major mode of reproduction is sexual or asexual, right? And what is the phylogenetic relationship, how an organism is related to the other organisms based on its evolutionary history, right? So evolution we have to consider, reproduction we have to consider, mode of nutrition we have to consider, the body organization or thallus organization we have to consider, and the basic thing, that is what is the kind of cell, so cell structure we have to consider. And in the table they even have given the cell wall, whether it is present or absent. If present, what is the chemical composition of that cell wall. So definitely we have to read R.H. Whitaker classification system in detail. We have to study all the kingdoms separately. That's why we will not take up much time here. You have to remember these five different you can say characters on the basis of which he divided all the living organisms. This is very important. Now Now this is the thing which is more advanced than five kingdom classification. Here there is no problem, it's not having any kind of drawback like the previous classification systems which I have mentioned, right? What was the drawback of these classification system that we got this classification system? So here I have mentioned the drawbacks, right? If you consider this Plantae and Animalia in two kingdom classification, can you see here bacteria and these all eukaryotic organisms are present together? Can you see in both? Here you have bacteria and here you have written mycoplasma, right? It means prokaryotes and eukaryotes are present together in kingdoms. That is not correct, not appropriate. Unicellular and multicellular organisms are present together, right? That is also not correct. Cell wall composition is not similar in the members. Somewhere cell wall is made up of peptidoglycan, somewhere it is made up of silica, somewhere it is made up of cellulose, somewhere it is made up of chitin, right? So chemical composition of the cell wall varies. That is not correct, right? And the mode of nutrition also varies. If you consider bacteria, you will see plenty of mode of nutrition here, right? Some can be saprophytic, some can be parasitic, some can be symbiotic, some can be autotrophic, right? In protist also you have same condition, right? Whereas fungi is exclusively heterotrophic and Plantae is completely autotrophic with a few exceptions which are like parasitic plants, right? Same goes here. So mode of nutrition is also not common in between the organisms. These were the drawbacks which we got in two kingdom classification system. Also some of them were removed in three kingdom classification, but some still exist. The four system came, right? After four kingdom classification, what happened? Five kingdom classification has removed all these problems. It has removed all the drawbacks, and it was a perfect classification system. But to make it more comprehensive, to make it more better, one more classification system came. It is called three domain classification or a six kingdom classification, right? Here no new kingdom is added, right? Here only you have to remember that kingdom Monera which was present in RH Whittaker's classification. How many kingdoms were there here? Here five kingdoms were there, right? Starting from kingdom Monera, Protista, fungi, Plantae and Animalia. These five kingdom classification was there. In this, the very first kingdom, that is kingdom Monera, was further divided into two, that was Archaea and Eubacteria, right? So Archaea and Eubacteria, these are two, you can say, new kingdoms which have come from the division of kingdom Monera, right? And then rest all the four kingdoms were clubbed under a new category that is called Eukarya. So we got three domains of life, okay? Archaea, Eubacteria and Eukarya, right? Or we can say total six kingdoms, right? Archaea, Eubacteria, Protista, fungi, Plantae and Animalia, right? This is also called six kingdom classification. They will always ask you which is the kingdom which got divided and which gave rise to two more kingdoms, right? So in that case, you have to remember kingdom Monera was split and it has divided and made two more kingdoms, Archaea and Eubacteria. Understood? Okay. Now, first one was kingdom Monera. So the most important points about kingdom Monera I have written here. We have to read this, right? So see, if you want to find where the members of this kingdom you will get, so they are cosmopolitan. Everywhere they will be present, in air, in soil, right, in water, right? If you take a handful of soil, thousands of bacteria will be there. So they are cosmopolitan. Plus you will find some of them in extreme conditions, maybe extreme saline conditions, right? Maybe anaerobic conditions, or maybe in very acidic conditions, very high temperature conditions where normal bacterial cell cannot survive. So along with the cosmopolitan distribution, they are also there in extreme conditions. Now if you will see them, their structure is very simple. Maybe you remember, you have studied the cell, the unit of life chapter, right? And there you have revised that the structure is so simple, right? As compared to eukaryotic highly organized cell, here you have very simplified structures. You don't even have a nucleus, right? So it's very simple structure, but along with that, it is showing us complex behavior. In different type of bacteria, you will see different type of metabolic pathways working, right? So they will always say that this is that particular kingdom where metabolic diversity is maximum. Okay, it yet it looks very simple, right? But still you will see that its behavior is very complex. You will see that in this particular kingdom, okay, bacteria will show you maximum metabolic diversity, because some of the bacteria will be doing some reactions, other bacteria will be doing another set of reactions, right? Some may be autotrophic. In that also you have categories: chemoautotrophs, photoautotrophs, right? uh, Aerobic, anaerobic, facultative anaerobe, facultative aerobe. So many categories are there. So as a whole, this group shows maximum metabolic diversity, simple in structure but highly complicated in behavior, right? Very, very important. Now nutrition, it can do photosynthesis, right? Or it can produce its own food by breaking some inorganic chemical compounds, so it can act like a chemosynthetic organism also. So in autotrophic condition we have two: we have chemoautotroph and we have photoautotroph. I'll tell you in the next slide. Heterotrophic, right? They can be parasites, disease causing. They can be symbiotic, right? They will be helping any other organism in that interaction, and they will get some benefit out of that, right? Just like nitrogen fixing bacteria, remember. Or they can be saprophytic decomposers, right? And here you can see their shape can be spherical. If they are spherical in shape, then you will call them as coccus. If they are rod shaped, you will call them as bacillus. If they are comma shaped, you will call it as vibrio, and if it is spiral in shape, you will call it as spirillum or spirilla. Here you can see this is a diagram given in NCERT. They can ask a question. Coccus always remains non-motile, no flagella at all. Bacillus may have, may not have flagella. You can see they have shown us a spore also. What is a spore? This is called endospore, right? A spore produced by bacteria, highly tough and resistant structure to avoid unfavorable condition, right? You can see so many flagella are present on both the ends of this spirillum bacteria. And you can see vibrio or vibrium, where you can see comma shaped bacteria. Here also you have flagella, right? So you can see this is NCERT diagram. You have to be very careful. You have to remember their names. You have to remember spore is present here, flagella is present, which bacteria does not have flagella at all, everything, right? Now I added one more image, which is from the last part of this kingdom monera, right? Here I have shown you a bacteria which is doing reproduction, and this reproduction is a asexual type of reproduction called binary fission. You can see the genetic material divided, entire cell has been divided, about to divide, or I can say, right? So it's a bacteria which is undergoing division, okay, dividing bacteria. Now, this entire kingdom Monera members are divided into two categories: Archaebacteria and Eubacteria. Primitive type of bacteria which are able to live in very harsh habitats or very extreme conditions where a normal bacteria will never survive, that are categorized under this category, that is called Archaebacteria, right? These are very primitive, right? So you will see them in those kind of conditions only, right? Based on where they are growing or where they are going to survive, based on that they are classified as halophiles, thermoacidophiles and methanogens, right? You have to remember this, right? They are present most harsh habitats. Now halophiles, they will grow in salty conditions, extremely salty conditions, right? You just imagine if a normal bacterial cell is put or kept in a solution which is having high salt concentration, what will happen, right? If as compared to the cytoplasm of the bacterial cell, the concentration of salt is more in the medium in which you are keeping that bacterial cell, what will happen, right? See, if you have this kind of condition, you have a bacterial cell kept here, right? And here what you have a salt concentration is very high here in this solution, right? What will happen? All the water will start coming out from a normal cell, right? And it will lead to the death of cell in some time, right? But a bacterial cell is able to survive in such an extreme condition, right? That bacterial cell is called halophile, which belongs to Archaebacteria, right? Thermoacidophiles. You know what are hot springs or hydrothermal vents, right? When under the sea bed, when the plate tectonics, they collide with each other, they form a crack open in the seabed under the water, right? And then what will happen? The boiling hot lava will touch the cold water which is present in the under the sea, right? And the water will be thrown away like steam, right? So temperature is more than 100 degrees Celsius there, right? Even more than boiling water temperature, right? And because a lot of minerals are present under the earth crust, so here you will see conditions are highly acidic. That's why it is called thermoacidophile, right? So these bacteria which are present in such condition, which are present in very extreme temperatures, very low pH or acidic pH, they are called thermoacidophiles, and you will be seeing them in hot springs, right? Methanogens. Methanogen is a methane producing bacteria which is present in the gut of ruminants. It is present in the anaerobic conditions, right? It does not require any oxygen to survive. It's an obligate anaerobe. So you have to remember these keywords. They will ask you which among the following does not grow in the presence of oxygen. It's an obligate anaerobe. So you have to remember it is methanogen. It is found in the guts of ruminants. Ruminants means these are the animals which have rumen in its digestive system, right? Like cattles, right? Like cow, buffalo, all of them, they have a particular extra organ in their body that is called rumen, right? Where the undigested cellulose will be present, and in that methanogen also is present, because methanogen help in the digestion of that cellulose, and it will not work in it will not work in aerobic conditions, right? And these methanogens are very useful for us because they will be producing lot of methane, which is a biogas, right? So we can use them for production of biogas because they are present in the cow dung, right? We can collect the cow dung, already methanogen is present there. We can feed our biogas treatment plant with this, mix the cow dung in water, make a slurry, add it to the organic waste which we have kept in the biogas treatment plant, and then it can act like an inoculum of bacteria, and it can do anaerobic digestion there. It can release lot of methane gas. We can use that gas for several purposes, right? So you have to remember. Now the question should be coming in your mind, why these bacteria are able to survive in these harsh conditions. Some are in salt areas, some are in hot springs, some are surviving without oxygen, right? So you have to remember this is possible because of difference in cell wall composition. NCRT mate is written as cell wall composition. Okay, along with that, you have to remember cell membrane also has some differences, and as a result, the archaebacteria is going to survive in extreme condition. What is the difference here? If you will see the cell wall ka composition in normal bacteria, it is definitely in this chapter they have mentioned that in the members of kingdom Monera, you have a cell wall which is made up of polysaccharide, right, and amino acids, right? They have written it like this in the table, right? RH Whittaker table. Now in eubacteria, if I write here instead of this bacteria, if I write here eubacteria, that will be more comprehensive. If I write eubacteria here, okay, in eubacteria the cell wall is made up of what? It is made up of peptidoglycan. What is the meaning of peptidoglycan? Peptidoglycan may you have a repeating unit NAG and NAM. So you have amino acids, you have these sugars, right? But in case of archaebacteria, in case of archaebacteria, you have this peptidoglycan is also called murin, right? So here you have pseudomurin. You don't have murin here, you have pseudomurin, and it is having a repeating sugar which is called NAG and NAT. Can you see it is having difference in the cell wall? It is not having the same chemical here. It is having different chemical. That's why it is able to survive. Another thing, what about cell wall? Sorry, what about cell membrane? So if you will see cell membrane here, this is about cell wall, right? If you will see cell membrane is having branch chain lipid. Okay, usually we have no branching in the fatty acid chain in the lipid molecule. Here you have branch chain in lipid, right? Because of these two characters, the archaebacteria ka outer structure, that is having cell wall as well as plasma membrane, it becomes very you can say strong and tough, and as a result these organisms can survive in extreme conditions, whereas a normal bacteria does not have this, right? In archaebacteria only you have these two characters. Based on the differences in the cell wall and plasma membrane, they are able to survive in extreme harsh conditions. You have to remember this. Now let's talk about eubacteria, right? If you remove the primitive group of bacteria, that is archaebacteria, then what is left? Then NCRT is saying that then there are certain bacteria which are left, which are called eubacteria, under which you have to study some of the heterotrophic bacteria. Okay, you have to study chemoautotrophs, you have to study photoautotrophs, which you know by the name of cyanobacteria or blue algae. So blue green algae, sorry. Here you have eubacteria. In the eubacteria, you have to remember you have rigid cell wall, right? You have flagella, right? And if the bacteria is motile, then only we have flagella, right? If the bacteria is non-motile, then no flagella, it will be lagged. So here they have mentioned flagella should be there, cell wall should be there. Now this cell wall should be of which particular chemical? Please tell me. If it is a eubacteria ka cell wall, it must be having NAG and NAM, right? Now see, if I talk about chemoautotrophs, we have divided all the bacteria here on the basis of the mode of nutrition, right? So autotrophic bacteria can be divided under two categories, chemoautotroph and photoautotroph. If you remember, chemoautotroph or chemosynthetic bacteria are that group of bacteria which will make their food, right? They will make their food. That's why they are like autotrophic bacteria, right? Another name of this chemosynthetic bacteria I'll write here, that is called chemoautotroph. Okay, either you can call it as chemosynthetic or you can call it as chemoautotroph. So if you are calling them as autotrophs, right, so it must be making food, right? If it is making food, then for preparing or synthesizing food, there must be source of energy, right? If without energy, can you prepare food? No. So that energy is coming from where? That energy is coming from the oxidation of certain inorganic chemical compound. What I said, the energy for preparing the food is coming from the oxidation of certain inorganic chemical compound. This group of bacteria will take the chemical compound, it will break the bonds. When the bonds are broken, energy is released. That energy this bacteria is going to use for preparing its food. That is why they are called chemoautotroph. For making the food or preparing the food, they are getting the energy from the oxidation of chemical compound, right? Okay, which kind of chemical compounds they can break? They can use certain nitrates, nitrites, ammonia, right? They can use all of these chemical compounds, inorganic chemical compounds, start breaking them and get the energy to make their food, right? Now these bacteria also help in the recycling of certain minerals, like like nitrogen, right? Like you can say phosphorus, okay, iron and sulfur. These all things will be, let's suppose any sulfur containing compound is there, bacteria is breaking that compound. So bacteria is just breaking the bond to get ATP, right? Bacteria is not interested to take up sulfur, right? So sulfur will be again recycled to the soil or not. Or let's suppose ammonia is there. Ammonia is the particular chemical which a bacteria is breaking. So what will happen? Ammonia may, when the bacteria will be breaking that, the bonds will be broken, right? So if the bonds are broken, then energy will be released. Bacteria will be taking that energy, right, to make its food. Bacteria is not interested in taking up the nitrogen. So nitrogen will get back to the soil or not. So it helps in recycling of these particular, you can say, chemicals, right? Now you understood chemoautotrophic bacteria. Can you tell me which bacteria should be categorized as chemoautotrophic bacteria? Can I say nitrogen? Can I say that those bacteria which are involved in nitrification, okay, which are called nitrifying bacteria. They can be categorized as the bacteria which are following this type of nutrition. Nitrifying bacteria, remember, which will convert ammonia into nitrite and nitrite into nitrate, right? uh, okay, understood. So this group of bacteria you have to remember. Now, what is photoautotrophic bacteria? Photoautotrophic bacteria is also called cyanobacteria here, right? Also called blue-green algae, right? If this bacteria is doing photosynthesis, definitely it requires a pigment. So just like green plants, it also has the pigment, green pigment, that is called chlorophyll A, the most universal pigment for photosynthesis. I think you have remember, you have studied photosynthesis chapter, there it is given. Now, the members of cyanobacteria, they are unicellular, they are single cells, unicellular. They can form colonies, right? You can see in the figure also. This is what, this is the filament of nostoc. Okay, this single cell is one bacterial cell. They all live together, all these filaments make colonies, right? uh, okay, they can live in filamentous form like this, right? All the bacteria can get aggregated together like colonies, right? Or they can exist simply as one single cell as unicellular bacteria. Now you can find blue-green algae in both sea, water, so marine conditions, as well as in the terrestrial conditions. Now colonies are, can you see this? Let's suppose this is one filament, or let's suppose we have a bacterial colony of this filament, right? So in this filament, can you see a hazy blue color outlining is given? This is what, this is a slimy layer which is called mucilaginous sheath, right? This mucilaginous sheath or a gelatinous sheath is surrounding the filaments of nostoc, or surrounding the filaments or colonies of any kind of blue green algae, right? It is there so that it can help the blue green algae from preventing desiccation or extreme dryness, right? And it also will protect the individual cells from the water currents. The cells will not get damaged, right? Now, you will see that these blue green algae will often form blooms in the water bodies, right? So in the polluted water bodies, you will see blue green algae will grow very rapidly, right? In the polluted water body, you have lot of phosphorus, and you know that nitrogen, phosphorus and potassium is very essential for the growth of any kind of cell, right? Even in plant it is most important, for other cells also it is important. So bacteria has, as soon as any detergent or any other kind of pollution is there in a water body, you will see very quickly blue green algae cells will increase there, they will multiply. So they forms blooms in water body, right? Mostly polluted. Then nitrogen fixing bacteria fix atmospheric nitrogen in specialized cells which are called heterosis. So one of the most important function of this blue green algae is that it will do nitrogen fixation. Remember, nitrogen fixation it will do. What is the meaning of nitrogen fixation? Can you tell me? This ammonia NH, sorry, this N2 gas is converted into ammonia within the cell, right? This is called nitrogen fixation. Okay, this is not done by this bacteria. Please remember, nitrification is the next step. When this is converted into NO3 minus, and this is converted into NO2 minus, that is nitrate and nitrite, right? This step is done by nitrifying bacteria like Nitrosomonas, Nitrococcus, Nitrobacter, Nitrocystis, right? And this step is done by any kind of heterotrophic bacteria, maybe, or maybe here in blue green algae also you can consider it, right? This is also fixing nitrogen, right? So this step will be done by blue green algae or maybe some heterotrophic bacteria like Rhizobium, right? And here you can see it is done by anabin or nostoc, right? So this step is called nitrogen fixation. The rest two things are called nitrification. Can I say the entire process of nitrogen fixation, right, can be achieved by both autotrophic and heterotrophic bacteria? We can say this, right? Okay. So you have to remember, so this first step will be carried by this bacteria. Not in all the cells of this blue-green algae you can do this step. For this step, you have to remember oxygen is very much a problem. So oxygen is not required at all. Conditions must be anaerobic here. For anaerobic conditions, all the cells, they cannot help to maintain it, right? So what you have to remember, in a filament of blue green algae, here example given is nostoc, you have to remember there are two type of cells. One specialized cell which is having a thick cell wall, in which one enzyme is present, right, which is responsible for nitrogen fixation, nitrogenase enzyme. That enzyme is also present in other cells, but because the conditions are aerobic in other cells, that enzyme cannot function, right? So for nitrogen fixation, here you have one enzyme that is called nitrogenase. Problem with this nitrogenase enzyme will work only in those conditions where you have to remember conditions are oxygen devoid or anaerobic, right? Anaerobic, right? Once the conditions are anaerobic, nitrogenase will become active. And once the nitrogenase is active, it will start catalyzing this reaction. Nitrogen gas will be converted into ammonia. For this, oxygen is not at all required, right? This will be achieved by having some modifications in a particular type of cell that is called heterocyst. What are those modifications? In this heterocyst cell, you have to remember there are two types of modification. The cell wall is very thick, right? It's having a thick cell wall, right? And you have to remember that it is having anaerobic condition, okay, because oxygen cannot enter. Another thing, if the bacteria is doing photosynthesis, you have studied photosynthesis, there are two photosystem, right? That is called ps2, ps1. Which is the photosystem which releases oxygen gas, right, by photolysis of water? That is photosystem 2. So here photosystem 2 is completely absent. Because of these characters, heterocyst can never ever make oxygen because pos2 is not there. Another thing, that cell wall is very thick, so it is impervious to any kind of gas. Oxygen will never enter from outside, so conditions will become anaerobic here. Nitrogenase enzyme will get activated. It will start converting nitrogen into ammonia. This is called nitrogen fixation. For this, which is the specialized cell which is present in nostoc, it is called heterocyst. Understood? And the other cells are normal vegetative cells, where what will happen? Carbon dioxide fixation, right? These cells are called vegetative cell. In this cell, what is happening? In this cell, CO2 fixation is happening, right? But in heterocyst, what is happening? Nitrogen fixation. So can I say blue green algae is capable to do N2 as well as CO2 fixation, but not in the same cell? Heterocyst is a special cell because it is having these characters, because of which the conditions remain anaerobic, and a nitrogenase enzyme can activate, it can catalyze this reaction, and it can easily do nitrogen fixation. Whereas the other cells, which are vegetative cells of this filament, they will be having ps2, ps1 both. They will be helping in photosynthesis or carbon dioxide fixation. Now you have to remember the name of these two blue-green algae, nostoc and anabina, which is given in this chapter. Where else you have studied about blue-green algae examples? Do you remember? There is a chapter called microbes in human welfare, right? In that they have mentioned that this can act like a biofertilizer, right? Because it enriches the particular soil with nitrogen, right? So there also you have examples of same nostoc and anabina, right? So I wrote here already that requires anaerobic conditions for nitrogen fixation, right? Now we have another type of bacteria which are heterotrophic bacteria. NCRT has written about heterotrophs also, that mostly the heterotrophic bacteria which belongs to kingdom Monera are decomposers. Imagine if the bacteria is not there. Bacteria means as a group I'm saying, right? It's a plural. So bacteria is a decomposer, right? If these decomposers are not there, what will happen? Earth will be full of dead bodies only, right? So it acts like a decomposer. What are the uses of heterotrophic bacteria? It is used in the formation of curd. Can you tell me which is that bacteria which is curd making bacteria? Remember lactobacillus? Lactobacillus, or there is a group of bacteria which is called lactic acid bacteria, right? These are the bacteria which help in making curd. Then many bacteria, they can provide us antibiotic for killing any other kind of microbe, right? They will be helping in nitrogen fixation in leguminous plants, so they are present in the root nodules, right? And some are pathogenic also. This is the benefits of bacteria, and here they have mentioned they can cause disease and damage as well. There are so many bacterial diseases, and they have mentioned some of the plant diseases and some of the animal diseases. Cholera, which is caused by Vibrio cholerae. Typhoid, which is called by Salmonella typhi, right? Tetanus, right? Clostridium tetani, and citrus canker, Xanthomonas citri, right? Citrus canker is there in lemons, in oranges, in citrus fruits, and these all are animal diseases, right? Then they have given one more type of member of kingdom Mona, that is mycoplasma. About mycoplasma, you have studied in cell the unit of life also. Please tell me, is this the smallest living cell? If yes, what is the size of this cell? Please tell me what is the size here. Size is very, very less, extremely less, right? It is how much? It is 0.3 micrometer. This is the size of this mycoplasma they have given in cell unit of life. Can you tell me what is the type of mycoplasma which is given in cell unit of life? It is PPLO, pleuropneumonia like organism, right? That is also a type of mycoplasma. Uska size is what? PPLO is a type of mycoplasma. Iska size they have given in NCRT is 0.1 micrometer, right? Okay, this is the particular bacteria which can survive without oxygen. Understood? It can survive without oxygen. So you have to remember it can live without oxygen, right? Okay. They will give this statement to you to they can write it cannot live without oxygen. No, it can live without oxygen, right? Then it causes both diseases in plants as well as in animals. Animals mein to this PPLO is causing this pleuropneumonia, right? And in plants, it is causing one disease which is not in NCRT, which is called witch's broom. Plant will appear like a broom of a witch, right? So this you have to remember about mycoplasma also. Now kingdom Monera is completed, right? We have a very big kingdom, that is kingdom Protista, but we have compiled every important point here. You can see kingdom Protista is divided on the basis of the mode of nutrition, right? In this kingdom you don't have similar mode of nutrition in all. You have different mode of nutritions here. Just say for example, if you consider chrysophytes and dinoflagellates as well as euglenoids, you will see mostly you will see that if I will uh, talk about this chrysophytes and dinoflagellate. Chrysophyte and dinoflagellates are purely exclusively photoautotrophic, right? They will do photosynthesis, right? Whereas the third one, euglenoids, in euglenoids you have to remember they are photosynthetic. That is true, they will be doing photosynthesis, but sometimes they will switch to another mode of nutrition, that is heterotrophic, right? So they can be photosynthetic, but at times they will start behaving like heterotroph. So this is called mixotrophic mode of nutrition. And then slime molds, protozoans, right? They are all what? They are completely they are heterotrophic, right? So this you have to remember, this is a continuation only for this, right? Understood? So this category we have to remember, this is the fourth one, this is the fifth one. These two are completely heterotrophic, these two are phototrophic, and this is what? This is showing us mix of both. Understood? On the basis of this only they have divided. Let's see where we are going to find chrysophytes. NCRT says that you are going to find chrysophyte both in freshwater and marine water. Chrysophyte is a group where you have two types of organisms as per NCRT, diatoms and desmate. Desmate is also called golden algae, right? So two types of organisms are there here, diatoms and desmates are golden algae. Now you have to remember they have mentioned that there is no motile stage in chrysophyte. Chrysophytes don't have any flagella, they lack flagella, right? Even that point was there in blue green algae also, right? Nostoc, Anabina, they don't have any flagella, right? Chrysophytes may also, you have to remember, they also don't have any flagella. Flagella is absent, right? So they will move passively with the water current, right? Now you have to remember that they do have cell wall, and their cell wall is made up of silica, which is very tough and resistant material. It's made up of silica, it contains silica, right? Then if you take if you see the cell wall is not continuous. Cell wall is in a form of two overlapping shells. So this is one shell, this is another shell, they are kept like this, right? Like a soap box you have seen, right? It's having two you can say parts. So like those two parts which will you can say overlap each other, like that they have their cell wall. So it is made up of thin overlapping two shells, right? And they appear like soap box or soap case. And you have to remember, because of the silica, which is very tough material, it is indestructible in nature. So cell wall of chrysophytes is indestructible, it cannot get degraded, right? It will not get you can say uh, destructed, right? So what will happen once all the diatoms, they will complete their lifespan, they will die. They are simple one cell structure only, right? They will die only. So what will happen after the death? The cell wall material will keep on depositing under the seabed, right? And that forms a powderish white color material that is called diatomaceous earth, right? Why this diatomaceous earth is important for us? It will take so many years to form some amount of diatomaceous earth, right? Because it's a like single cell organism only. All the protists are having only one single cell, which are eukaryotic, right? So this diatomaceous earth is very important because it is gritty in nature. Its character, it is rough and gritty. So you can use it for polishing, right? For polishing or for filtration of oils and syrups, right? These are the two things. We can use it for polishing, we can use it for filtering oil and syrup. We can prepare filters by this, right? So it is gritty in texture. It will take many years for diatomaceous earth to form. What is diatomaceous earth? It is made up of silica, right? Where silica is present? In the cell wall of diatoms, right? Is it destructible or indestructible? It is indestructible, right? Cells cannot move by flagella because they lack flagella. They can move passively only. Their cell wall is in the form of a soap case, right? Now you have to remember, diatoms are chief producers of oceans. What's the meaning of chief producers of ocean? See, the single cell organisms which are eukaryotic, which are present in the oceans, which are doing photosynthesis, they are called planktons, right? Remember, in the producer level, in the aquatic ecosystem, in the food chain, what is present at the first trophic level? That is called plankton, right? And the plankton which will be doing photosynthesis, that is specifically called phytoplankton. So you can call that these are actually the phytoplanktons. Phytoplankton does not have any flagella. They are non-motile green photosynthetic cells which are present in the water, right? So these planktons are the chief producers. Diatoms are the chief producers of ocean. You have to remember this, right? These are the important characters about this diatoms, or you can say chrysophytes, right? Now dinoflagellate, name itself is telling you dinoflagellate means di means two, flagellate means flagella, so it's having two flagella. Dinoflagellates have two flagella. Most of them are present in sea, so they are mostly marine in nature. They are also photosynthetic, they have photosynthetic pigment, so they can appear yellow, green, brown, blue, red, depending upon the relative concentration of the pigments, right? They have different types of pigment, right? In which again the main photosynthetic pigment is chlorophyll A only in both of them, right? Then here you have to remember their cell wall is stiff, right? It is made up of cellulosic plates. Stiff cellulosic plates are present in the cell wall, right? That's why it is also called as armored dinoflagellate sometimes, right? On the outer surface, on the surface of the cell, you will see this thick uh, stiff cellulose plates are present. Two flagella are present, right? One is called longitudinal, another is called transverse. I'll show you one image, right? Here they have mentioned dinoflagellates. If I want to draw its easy structure, so you can consider dinoflagellate will somewhat appear like this. Okay, there will be a groove like this. Okay, now this groove is called transverse groove. Okay, can you see a depression type of or you can say a groove type of structure here? This is called longitudinal groove. Okay, two grooves are there. Now one flagella, right? Okay, you can see two flagellas are there, right? One flagella is present here, another flagella is present here, right? This flagella is which is present in the longitudinal groove is called longitudinal flagella. It's actually thin and very long, right? Other flagella which is present in the transverse groove, this is called transverse flagella. And this flagella is ribbon like small as compared to the other one. So can I say two different types of flagella are present? This condition, if you remember, it is also called heterocont, right? Cont means flagella. So it's having two different type of flagella, longitudinal and transverse flagella present in the longitudinal and transverse groove. Even in the image also you can see they have shown this longer one, that is longitudinal, and transverse one will be present here, right? Okay. Now there is a specific type of dinoflagellate which is called goniolax, right? Which is having predominant red pigment, right? So here it is red in color because it is a single cell. It will divide by binary fission, right? And rapidly, very fastly, it will cover the entire water body into red color. That condition is called red tide. So goniolax causes red tide. It multiplies very fastly, right? Because it's a unicellular eukaryotic cell, right? So you have to remember this. Now we have euglenoids, right? These are photosynthetic groups, right? I told you these both are photosynthetic groups, right? Here you have this one, that is euglenoids. Euglenoids, may you have to remember, euglenoid remains in the fresh water. Mostly you will see them in stagnant water like pond water, right? Here you don't have any cell wall. Instead, one very flexible layer is present, that is called pellicle. Pellicle is proteinaceous in nature. It is made up of protein and it provides flexibility, right? So it gives flexible character, right? And it's protein in nature, right? Now this also have two flagella, one is short, one is long. So you can say that yes, here also heterocont condition is there. Can you see, right? This near the you can say anterior part of the cell, you can see two flagellas are there. One is long, one is short flagella, right? So two different types of flagella exist here also. Mode of nutrition in euglenoids is mixotrophic. When the sunlight is present, it will act like a photoautotroph. When the sunlight is absent, it will act like a predator. It will be predating on some small marine form, like not marine, sorry, small cells or organisms which are present in the you can say which are smaller than euglenoid. They will kill it and it will eat it, right? Eat in sense, it will take it inside its body and use it as source of nutrition. Euglena is the one most important thing. It is having exact same pigment combination which is there in Higher plants. Higher plants have the main photosynthetic pigments as chlorophyll and A and chlorophyll B. Okay, same as plants. If you will see higher plants, the pigment combination, it is chlorophyll A and B. Same pigment combination is present in euglenoids as well. Now we have slime molds. Slime molds are heterotrophic organisms, right? Here so you have mixotrophic, here they are completely heterotrophic. In heterotrophic also, which kind? This is saprophytic, right? They'll be doing saprophytic nutrition. Now slime mold is a particular category where you have to remember, here also you don't have any cell wall, right? Here there are two different stages. One is called plasmodium stage, another is called fruiting body, right? One is plasmodium, another is called fruiting body, right? So what is happening here? Two stages are there, plasmodium and fruiting body. You have to be very careful, right? See, what is the work of this particular slime mold? Slime molds means small single cells are present, which are actually releasing the enzymes outside, just like fungus, right? Breaking the complex organic material, which can include leaf litter, dead wooden logs and all, right? It will break it down and it will absorb the simple nutrients within it, right? Just like fungus. So this is the saprophytic type of nutrition which it is going to perform. So let's suppose so many decaying twigs, decaying leaves, right? Or all this organic matter is present, right? What this slime mold will do, different slime mold cells, they will aggregate together and they will form a multicellular structure, sorry, multinucleate structure, that is called plasmodium, right? So this multinucleate structure is called plasmodium. I think here there is an image, you can see here multiple nuclei are present in a single cytoplasm, right? This will happen how? Okay, let's suppose this was one cell of slime mold, this was another cell of slime mold, this was another cell of slime mold, this was another cell of slime mold. What happened? These all cells, they have got aggregated, right? All these cells got aggregated. यहाँ पे first step हो रहा है, that is aggregation, right? They aggregated so that they can increase their absorptive surface area, right? Up, just imagine surface area for absorption here is less, right? When they all will combine, the surface area will become more, right? So this aggregate, they became aggregated so that they can increase their absorptive surface area and they form a multinucleate structure, right? This is a multinucleate, or you can say cenocytic structure, which is called plasmodium. So plasmodium is a stage of slime mold, right? Which is, I should not underline it. This is only there for feeding, right? When the conditions are favorable, when the conditions are favorable, favorable means enough food is available, right? Lot of organic matter is present, lot of decaying log, what a tree log and leaf litter, everything is present. So what will happen? All the slime mold cells will come and aggregate together. After aggregation, they will make something like this, which is a multinucleate cenocytic structure, which is called plasmodium. Its work is only to spread up to several feet and keeping on absorbing the nutrients from this organic matter, right? Understood? So it grows over several feet and just it will eat, right? By absorbing the nutrition from this dead and decaying organic matter, it will break it down, it will engulf all the nutrients, right? Then what will happen? Food will be finished, right? After some time, right? Now the conditions are unfavorable. Favorable condition means food is present, right? This plasmodium will grow only when conditions are favorable. When the conditions are unfavorable, this plasmodium will start differentiating fruiting bodies, right? So what is fruiting body? On this multinucleate structure, you will see some fruiting bodies will appear like this, right? This differentiation will occur and you will see fruiting bodies appearing from this structure, right? What is this fruiting body? This fruiting body will be having, at the tips you will see there will be formation of small spores, Okay, this is what? These are the spores. This is the fruiting body. On the tip of the fruiting body, you will see lot of spores will be formed, right? And when this particular, you can say, structure will rupture, the tip of the fruiting body which is having lot of spores, it will rupture, all the spores will be liberated, right? And then all these spores are dispersed by air current, they will be carried away by air, and wherever they will fall, new slime molds will be appeared, right? So this you have to remember. And one more important thing, this is the group which is producing spores with true cell wall. What's the meaning of true wall? It is cellulosic in nature. So if they ask a question, which is that particular group from kingdom Protista, which is sharing common characters between kingdom Animalia, kingdom Plantae, and kingdom fungi. So what you have to remember? You have to remember this. Can you see? This is this plasmodium stage made. There is no cell wall. Cell wall is absent. This is a character just like animals, right? Can you see that spore cell that the cell wall of the spore is having cellulose? This is plant like character, right? And fruiting body formation, saprophytic mode of nutrition, this is the character like fungus or not, right? So three different things you can see in one particular member of kingdom Protista, that is slime mold, where you have to remember all these things, right? When enough food is present, conditions are good and happy, favorable, then this they will do, slime molds will aggregate, they will form a feeding stage that is called plasmodium, which can grow up to several feet, right? Once the conditions are changed to unfavorable, food is finished, what they will do, fruiting body will start getting produced on the this slime mold, right? Sorry, on this plasmodium stage, fruiting bodies will appear. At the tip of the fruiting body, spores will be formed. Spores will be having true wall. When they will be you can say dispersed, they will be dispersed by wind, right? Now next one is protozoans. So in protozoans you have to remember, pro means primitive, zoan mean animal. We say that these are the primitive forms which have may given rise to animals, right? Because the mode of nutrition is heterotrophic here, right? They are heterotrophic, they can be predators or they can be parasites, right? Two mode of nutritions are there, predators or parasite. Whereas in amoeba you will see that amoeba, flagellated protozoan, ciliated protozoan, and sporozoans, there are four different categories in which we have divided them, right? Here you can see this amoeboid, flagellated, okay, ciliated, and here I have written sporozoans. Four are there. They all are divided on the basis of their locomotory structure. If the locomotory structure is pseudopodia, they are clubbed as amoeboid. If it is flagella, as flagellated. If cilia is there for locomotion, ciliated. And if there is no motile structure, right, I am writing here, they are non-motile, they don't produce any kind of locomotory structure, so they are considered as sporozoans. Okay, now you have to remember some few points in all. Just like for example, in amoeboid protozoans, you have to remember some of the members of amoeboid protozoan which are marine, they have silica shells on their body, right? So silica shell is present on amoeboid protozoan. They don't have any cell wall. This is not cell wall. This is just a shell which is made by silica, right? And there are two types which are given in NCERT, amoeba and entamoeba. You have to remember. Flagellated may you have to remember that they are mostly parasitic, they will be causing diseases. Even some of the members of amoeboid protozoan like entamoeba also causes diseases. They are also parasitic. Here also they are free living or they are parasitic. They will cause a disease called sleeping sickness, and the type of protozoan which is responsible for that is trypanosoma. Trypanosoma is a flagellated protozoan which causes sleeping sickness. Then you have ciliated protozoan, where you have example as paramecium, which is having thousands of cilia beating together, right? So that the water laden food can enter in a specific cavity which is called gullet, right? So you have to remember here, gullet is there where actively thousands of cilia are beating, so the food which is present mixed in water will enter inside the cell. They also have contractile vacuole, right? Protozoans have contractile vacuole for what? For removal of all the waste which is present in the cell. They have a specific opening called gullet for entry of things inside the cell, like food particles and all. And for removing the waste outside, they have another opening that is called contractile vacuole. Then you have a non-motile type of protist, which are called sporozoans. They have infectious spore like stage, which is called sporozoite. Okay, this sporozoite is what? It is an infectious spore like stage which is present in one of the members of sporozoan, that is called plasmodium. Plasmodium is that particular malarial parasite. It causes malaria, right? So these are five different you can say uh, members which you have to study in kingdom protista. These you remember. Here you have cell wall of overlapping shells. Silica is present here. They are chief producers of oceans. They are floating passively, right? They you can say they will be making diatomaceous earth, right? It includes diatoms and desmate. Desmate is called golden algae. You can use it for polishing. Diatomaceous earth you can use for polishing, filtration of oil and syrup, because it is gritty in texture. Dinoflagellates, they have two flagella, so they will not move passively. They will be using flagella and move actively in the water. Same like this, euglenoids also have two flagella. This is photoautotroph. This is mixotrophic, right? Here colors have a range. It depends on the concentration of pigments. Green, brown, blue, red, yellow, every combination is possible. Here they are mostly green in color. They have a pigment combination just like higher plant, that is chlorophyll A and B, right? Here you have cell wall made up of stiff cellulosic plates on the outer surface. Here you don't have cell wall, you have a flexible layer which is proteinaceous, which is called pellicle, right? Here you have two flagella, one in longitudinal groove, which is called longitudinal flagella, which is longer, right? Another in transverse groove, which is transverse flagella, or you can say a ribbon shaped small flagella, right? Here there is a particular example, goniolax, which will multiply very rapidly and it will cause a condition called red tide, right? It is different from one more condition called red sea, that is caused by one blue green algae called Trichodesmium erythraeum. Here you have euglenoids, you have to remember it can act like a predator when sunlight is absent, okay, and it will be doing photosynthesis when sunlight is present. That's why mesotrophic mode of nutrition. On the other hand, slime modes, I told you that this is heterotrophic mode of nutrition, is just like fungus, right? When the food is present, conditions are favorable, all the slime modes will aggregate together. They will form a multinucleate synocytic structure called plasmodium. It can grow up to several feet and its work is only one, to feed upon the dead organic matter, right? Once the conditions are changed to unfavorable, food is finished, then this structure will start differentiating fruiting body. At the tip of fruiting body there will be spores. Spores are having cellulose containing true wall, right? Then the tip will rupture and you will see all the spores are liberated. They will be dispersed by air, right? Protozoans, they are having different categories based on what locomotory structure they have, right? Most of them are heterotrophic. They can be predator, they can be parasites. Amoeboid, silica shell is present. Flagellated, trypanosoma, sleeping sickness it will cause. Ciliated, paramecium is an example. It is having so many thousands of cilia beating together. They have two openings, one for entry of the food particle, that is called gullet, one for uh, exit of the waste material, that is called contractile vacuole, right? And then you have sporozoans, where you have malarial parasite, which is called plasmodium. This plasmodium and this plasmodium is absolutely different. In question, when they are writing P as capital, everything in italics in your NEET exam, you have to remember they are talking about this fellow. When they are just writing plasmodium, then they are talking about a stage which is there in slime mold. Plasmodium is a stage of slime mold, feeding stage, and plasmodium is also a name of malarial parasite if written in italics. It's a genus name, right? It causes malaria. And the spore of this particular plasmodium which causes malarial infection is sporozoite, right? No motile structures, right? This is a paramecium, right? You can see here, you have to remember. What if they give any question from here just in a form of an image and they will ask us to do, right? They'll give some characters to us. Now the extreme important group, that is kingdom fungi, right? We have completed Monera, Protista, now fungi. All the members here exclusively heterotrophic. They don't have any chlorophyll, but it does not mean they don't have any pigment, right? Please remember, pigments are present. Pigments are present. Okay, chlorophyll is absent. Are you able to understand? If they give a question that all the photosynthetic pigments are absent, false, right? Because you have seen this fungus can be of orange color, sometimes red color, yellow tinge is there, so it means they have pigments, but they don't have green pigment, right? Green pigment is very necessary for the photosynthesis. This is the main photosynthetic pigment, right? This is the main photosynthetic pigment. This pigment is not there, right? This pigment is absent. Understood. But other pigments are present. All the members of this kingdom are multicellular except a unicellular fungus that is called yeast, right? The cells of this kingdom all are haploid. They are carrying haploid cells. So these haploid cells, they are joined together to form a filamentous thin tube-like structure which is called hyphae, and this hyphae will make a dense network which is called mycelium. So let's suppose if I want to draw here, okay, if I draw a small part like this, can you see a tube like multicellular structure which I'm drawing here, right? This is a one cell. Can you see this cell is haploid? And can you see it is making a filament which is called hyphae? And then if I change this into one line only, you have to remember this is a complete structure, right? So it is making a dense network like this, which we see cottony mass appearing on fruits and all. This is what? This is called mycelium, right? So the thallus or the body here is of what type? It is mycelium. Thallus word we will use when nothing is differentiated, right? It is undifferentiated only. So mycelium, hyphae, cell, you understood how they are connected, right? One cell of fungus is haploid in nature, right? These cells connect to make a thin tubular structure which is very thin, which can be used for absorption of water, mineral and all, right? So this is hyphae, this is very thin tube like structure. And mycelium is what? This hyphae together forms a network which is called mycelium, right? So this you have to remember. Mode of nutrition, you can see it can be heterotrophic, so it can be saprophytic, decomposer, parasitic, right? Symbiotic, so many mode of nutritions are there. If you talk about reproduction, they will reproduce by asexual spores. Zoospores are there, sporangiospores are there, conidiospores are there, right? When you will see zoospores are actually motile spore like this. They have flagella, right? When you will talk about sporangiospores, these are non-motile spores, right? When you will talk about conidia, they are also non-motile spore, but the difference is these two are formed within a structure, right? Which is called zoosporangium and sporangium. But conidia is not formed inside any structure. So it's exogenous in origin. These are endogenous in origin, right? Then we have sexual spores as oospore, ascospores, and basidiospores. About them, we will be learning in which of the fungi, which type of spore is there. And the sexual reproduction in fungus is of very unique type, because here the sexual reproduction does not happen by normal method which we have studied in others. Here the first step in sexual reproduction is plasmogamy. Gametes are formed, gametes are fused, and only the protoplasm part of gametes are fusing, that is called plasmogamy. Next step may the nucleus part will fuse, that is called karyogamy. Sometimes karyogamy will occur very fastly, immediately, right? In lower fungi this happens, but in other fungi you will see that after plasmogamy there is a small duration lag. Nuclei will remain unfused. It will give rise to a stage called dikaryotic stage or dikaryo phase, right? Where you will see two unfused nuclei in a single cell, that is called dikaryotic stage or dikaryo phase. In after that the two nuclei will fuse, that is called karyogamy, and then in that diploid cell there will be meiosis, because fungal cells are haploid. You want to make haploid fungus, so at last you have to do meiosis in the diploid structure, right? So three steps are there: plasmogamy, fusion of protoplasm, okay, karyogamy, fusion of nuclei, and meiosis at last, so that you can make these sexual spores which are haploid, right? Now all the fungi are divided on the basis of what? This is a PYQ, they have already asked, right? So all the fungi are divided on the basis of morphology of mycelium, mode of spore formation, and fruiting body, right? What kind of fruiting body is present, what kind of asexual spore, sexual spore they are forming, and what is the kind of mycelium. Based on this, we have different types of fungi. In this chart you can see I have tried to compile most of the things together, right? So if you see their habitat which is given in NCERT, can you see Phycomycetes, Ascomycetes, Basidiomycetes and Deuteromycetes? These are the four different types of fungal classes which we have to study. Okay, habitat, if you see, almost everything is same with a few unique things given in NCERT. Because it's a fungi, definitely it will be present in the place where dead and decaying organic matter is present, because it is heterotrophic, right? So everywhere you can see, except Phycomycetes where they have mentioned aquatic also, no other fungi they have mentioned is aquatic. Here only they have mentioned Phycomycetes, some few members of Phycomycetes which are belonging to a primitive category, Oomycetes. Okay, Oomycetes, that is aquatic. Rest all members are terrestrial. If they are terrestrial, they can be found on moist, damp soil, or on decaying wood, right? Or as parasites to plant, right? Then here you can see they can be saprophytic, decomposers, parasitic, coprophilous. What's the meaning of coprophilous? Who will tell me? They will grow on cow dung. Okay, coprophilous means this fungi will grow on cow dung, right? Then basidiomycetes, mostly you will find them on the tree or wood, right? So they are present on soil, obviously they will be present on logs, wooden logs, tree stumps, right? They can cause diseases also like rust and smut. They are parasitic, right? And deuteromycetes, they are saprophytic. They are, majority of them are decomposers of dead and decaying organic matter, or decomposers of litter. If they are decomposing the organic matter or leaf litter, definitely they will be recycling the minerals back to the soil or not. So you have to remember this, very important. This was a PYQ, already they have given this, right? That which among the following fungi will be decomposer of litter, and they will help in what? So you have to remember, mineral recycling may this fungus will be providing maximum, right? They will also be responsible for minerals recycling because they are also decomposers, right? But this is contributing maximum, right? So you have to remember, right? Now this you understood. Now if you see the mycelium, remember the particular thallus organization of fungus is called mycelium, right? Mycelium is what? Network of hyphae. Hyphae is what? A thin tubular structure, right? So if you see mycelium here, mycelium will be unbranched. You will never see any kind of branching. And the mycelium is aseptate. What is the meaning of aseptate? Aseptate means there is no cross wall in between the, देखो, here if I have made this thin tube like structure, right? And here also, if let's suppose I will make a thin tube like structure, right? They both are looking different from each other or not? If you try to see them, right? In the very first one, what I'm going to do, I'm going to draw certain nucleus without any kind of septa or cross wall in between. So can you see, continuously nuclei are present in a single protoplasm or single cytoplasm. This kind of condition is called multinucleate. And another technical term which we use for fungi for the same condition is cynocytic, just like that plasmodium which we have covered, right? So this multinucleate cynocytic condition is present here, right? Here what is the condition? Here you can see septa is present, right? If I draw a structure like this, right, branching is also there. Okay, so this is what, this condition is called septate. You actually have these cross walls or cell walls, right? Can you tell me how we can get this condition done? If in the cell cycle, if karyokinesis is working normal and cytokinesis is failed, then you will get cynocytic condition. And if karyokinesis is followed by cytokinesis, during the mitosis you will get this normal condition, right? So you have to remember, in the first you can say member in fungus, okay, here only you have aseptate, Okay, this cyanocytic condition is also called aseptate. A means septa is absent. Okay, what is the meaning of septa? Septa means cross wall, right? Septa means cross wall or the cell wall which is present between the two nuclei, right? So it is aseptate, no branching. Here branching is there and it is septate. Can you see Esco, Basidio and Deutero all are following this condition, right? Now, if you talk about cell wall, when you will read sorry, when you will read fungus cell wall, they have mentioned here that they have cell wall. All of them they have cell wall, and they have mentioned that cell wall is made up of chitin. Okay, what is chitin? It is a homopolymer of NAG. Okay, it is a homopolymer of NAG plus polysaccharide. Some of the polysaccharides, other polysaccharides are also present. You check NCRT, they have given like this, right? Cell wall of algae is having chitin plus other polysaccharides. So I mentioned chitin here. Everyone is having chitin. An exception is there in phycomycetes. There is one group of phycomycetes where you have cellulose also, but it's not mentioned in NCRT, so we don't have to study that. Then asexual reproduction will be done by asexual spore, which will be formed by mitosis. So you can call them as mitospores as well, right? Understood? So if let's suppose that this is a particular fungal mycelium. In this fungal mycelium, there is a structure which is formed like this. And in this structure, mitosis is occurring, right? And it is forming certain spores which are this kind of spore, right? Motile, asexual, endogenous. Origin wise, they are endogenous because they are formed within a structure called zoosporangium. This is a specialized type of mycelium. The mycelium has become specialized to form this structure which is called zoosporangium. And in this zoosporangium, you have certain spores which are called zoospores. All these spores are haploid. They all are formed by mitosis. That's why you will also call them as asexual spores or mitospores, right? This kind of spores are found in phycomycetes, right? One more type of spore is there in phycomycetes. Let's suppose this is the fungal mycelium. This hyphae got specialized to form this structure, which is called this time as zoospora. Sorry, this is called sporangium only. This structure is called sporangium. In this sporangium, there are small spores which does not have any kind of flagella. These spores are actually non-motile, right? Non-motile asexual spores, right? These are asexual, right? And they are also endogenous. Okay, one more thing I will tell you here. There is one term used in NCRT. If any spore is motile, they have called it as planospore. Okay, if any spore is motile in nature, they have been called planospores. And if any spore is non-motile in nature, it does not have any flagella, so they have called it as aplanospores, right? Aplanospores. They have called this as aplano. So we can say either sporangiospores are aplanospores, right? Sporangiospores are aplanospores, or we can call them as non-motile spores, because at times in NCRT that word is used, so you should be familiar with this. When you are reading it, please read it. Sporangiospores are also called aplanospores because they are non-motile. Plano means motile, right? Understood? Both of these spores are found in phycomycetes. Can I say phycomycetes produces asexual spores which are endogenous in origin, which are covered by this covering zoosporangium and sporangium? Yes. All the spores of phycomycetes, all the asexual spores of phycomycetes, they are motile. That is wrong, right? So Here, can you understand this point? You have understood, right? Conidiospore, which is a another asexual spore, which you will see in, see, let's suppose here this is a specialized structure of hyphae, right? On this structure, I'm saying we have certain more hyphae specialization. So we have a condition like this. I'm drawing only two, right? On this we have small cells which are like this, elongated bottle shaped cells are there. Same will be there on this also. Here at the tip there will be mitosis which will be occurring, and we will be making asexual spores like this, right? These spores are called conidiospores, right? These spores are called conidiospores. They are non-motile. They are formed on a specialized structure which is called conidiophore. Okay, it's a specialized hyphae on which these conidia are born, and you can see the smallest is present here, the eldest is present here. So this is called basipetal arrangement, young at the base and old at the tip, right? So they are non-motile conidiospores which are found in two types of fungus. First is ascomycetes, second is deuteromycetes. In both of them you will get this asexual spore which is like this. And now can you see it is not covered by anything? So you have to remember, origin wise they are exogenous, they are not covered into any sporangium, they are exogenous, right? So you just see here we have mentioned same thing, that here we have conidiospores, here also we have conidiospore, this is exogenous, this is also exogenous. Now let's talk about sexual spore. So here the sexual spore is oospore and zygospore, name must be remembered, right? Here ascospores in ascomycetes, basidospores in basidiomycetes, and it is not yet reported for deuteromycetes. Sexual reproduction in deuteromycetes is not known. That's why they are also called as imperfect fungi. Imperfect fungi, also called as fungi imperfecti, right? What is the reason for this? Sexual reproduction not reported. You have to remember, you cannot say it is absent. Okay, we don't have any particular uh, you can say findings about their sexual reproduction. Sexual reproduction exists, but it is not reported to us, right? That is what we have to remember. They are called imperfect fungi. Perfect fungi, sexual stage, we know them, right? Iska sexual stage is which one? Ascospore it is going to produce, or basidospore it is going to produce, or zygospore or oospore. But here, what kind of spore is there? We don't have any idea. We have not find it yet, right? We have not found it yet. So we have to remember they are called imperfect fungi, right? Because sexual stage is not reported, never absent, right? That option, if it's there, it will be wrong. Okay, so asexual spore is present in all except basidiomycetes. They don't have asexual spore. They will reproduce by fragmentation, because they are filamentous. Hyphae can do fragmentation, right? Vegetatively it will reproduce. And sexual spore, everyone is having sexual spore, but we don't have which we don't have any idea which sexual spore is present, deuteromycetes. So these two things you have to remember here. So it is absent in basidiomycetes, asexual spore is absent, but in deuteromycetes sexual spore is not reported, right? Then fruiting body, it is not formed in phycomycetes. Yes, it is formed in asco and basidio, not reported here. In asco the name of fruiting body ascocarp, in basidio the name of fruiting body is basidiocarp, right? The sexual spore which they produce also having similar names, right? What is the place where meiosis will occur so that you can make sexual spore? Here it is zygote, right? Here it is ascus, right? It's a finger like structure here, and here it is basidium. I'll tell you about this sexual reproduction also very quickly, right? Just a minute. Okay, I'll write here. Let's talk about phycomycetes here. Okay, ascomycetes here, and basidiomycetes here. Okay, you have to remember, let's suppose this is the mycelium of phycomycetes, this is the mycelium of phycomycetes. Okay, they will be producing gametes. So this is, let's suppose, one gamete, this is another gamete, right? We don't have any male and female in fungus. We can have strains, positive strain, negative strain, like that, right? So two mycelium which are genetically different, right, they have produced gametes. Up the first step in sexual reproduction is plasmogamy, right? First step, you remember, first step is plasmogamy. So I'm writing here, it is plasmogamy. Second step in, you can say, sexual reproduction is karyogamy. So I'm writing karyogamy. Why I'm writing it together? Because here you have to remember, karyogamy is immediate. Just so as soon as the cytoplasm of the two gametes will fuse, at the same time the nucleus nuclei will also fuse, right? So here we have the first step, that is plasmogamy, karyogamy, second step, but karyogamy is immediate karyogamy. It will result in the formation of this diploid structure which is called zygote in NCERT, right? Up in this zygote there will be meiosis, the third step. When meiosis will take place, depending upon the type of member of phycomycetes, we will be producing sexual spores. The sexual spores are haploid, because in diploid, if you are doing meiosis, haploid cell will be formed. This spore can be of two types. Either it can be oospore or it can be a zygospore. Understood? One more thing I will tell you, I'll not write it, it's very confusing. The name of zygote is also same as the name of sexual spore, right? You can say the zygote in oomycetes is oospore. The name of zygote in phycomycetes ka zygomycetes is zygospore. Two types of phycomycetes are there. Okay, one is called oomycetes, another is called zygomycetes, right? Understood? So result of karyogamy, diploid cell in oomycetes is oospore, in zygomycetes it is zygospore. And the result of meiosis in this diploid zygote, which is having the same name as sexual spore, is oospore or zygospore, which are haploid. Matlab, name of sexual spore and name of zygote is exactly same, only ploidy is different. Only result of which condition it is, that you have to remember. If I'm saying that a diploid zygospore is formed in zygomycetes members of phycomycetes, right, then I'm talking about a diploid zygote which has to do meiosis. If I'm saying that we have zygospores formed after meiosis in diploid cell, which is called zygote, so you have to remember I'm talking about sexual spore, right? If you cannot remember this much at this point, you can just remember this is a diploid structure called zygote. No need to remember its name, right? It will undergo meiosis, it will form the spores. Okay, this is not given in NCERT. Then ascomycetes. Ascomycetes also two strains I am drawing here. Okay, and these two strains will be making gametes. Obviously they will be having sex organs, they will be making gametes, gametes will be formed, gametes will fuse, karyogamy will take place, sorry, plasmogamy will take place, right? After plasmogamy, you will see there will be one intervening stage which will be formed, which will be called as dikaryotic stage. This stage is called dikaryotic stage. Here you have two unfused nuclei. Here karyogamy is delayed, not immediate. Okay, but this is a short lived stage. I mean, This dikaryotic stage is short lived. For very small duration this is present. Understood? Now this particular cell will divide and it will form a structure called fruiting body. Okay, what is the name of fruiting body here? The name of the fruiting body of ascomycetes is ascocarp. This ascocarp appears like this. There are different forms of ascocarp depending upon their structure, right? On this fruiting body there are some small finger like projections which are formed, right? This is called ascus. So on ascocarp we have ascus, right? In this ascus only what will happen? Karyogamy will occur. Once karyogamy is occurring in the ascus, the cells will become diploid, right? Now in that only meiosis is going to take place. After karyogamy, meiosis will occur, right? And if meiosis is going to occur within this ascus, what will be formed? Ascospores. Okay, here you will see 1, 2, 3, 4, 5, 6, 7, 8. 8 ascospores are formed within one ascus. All the ascospores are formed by meiosis, so they all are haploid. Understood? And can I say that all these ascospores are endogenous in origin because they are formed within the sac-like structure called ascus or asci, right? Plural may it is called asci, and one singular one is called ascus. So you have to remember this is very different. Here you have to remember sex organs are present. Here also sex organs are present. That's why they are able to make gametes. Gametes are able to undergo plasmogamy. Here immediate karyogamy. Here dikaryotic stage is formed. After dikaryotic stage, here fruiting body is formed, which is called ascocarp. Here, once karyogamy has occurred, meiosis will take place, right? And it will directly form sexual spores. Here, delayed karyogamy will be followed by fruiting body formation. All the cells will be having this condition, n plus n. Then the two nuclei will fuse. Fruiting body may one structure is there that is called ascus. Within that ascus, karyogamy will take place, followed by meiosis, which will give rise to ascospores within this structure called ascus. Right? All the spores are endogenous in nature because they are formed within this sac-like structure called called ascus, right? Now here let's draw for basidiomycetes. The problem in basidiomycetes is that in basidiomycetes they don't have any sex organ. So sex organs here are absent. This is a problem. If sex organs are absent, gametes cannot be formed, right? Gametes cannot be formed. So there will be somatic fusion, somatic cells fusion, which is called somatogamy, right? Somatic cell or body cell or thallus cells will fuse, right? This will be cynocytic cell, septate cell, right? Septa is there. So the terminal cells of compatible mating type of the basidiomycetes, they will fuse. This kind of plasmogamy is called somatogamy. First step is completed. This special type of plasmogamy is called somatogamy. Here the body cells are fusing because the sex organs are absent. Right? This fungus is very, remember that this is not having any asexual spore and it does not have sex organs also, right? It will not do asexual reproduction, it will do vegetative reproduction, but it will do sexual reproduction by fusing its somatic cells. So plasmogamy will take place and this cell will change into again this kind of cell, n plus n, right? This cell is called again dikaryon or dikaryotic stage ka cell. Up this you have to remember is long lived stage here. Okay, Now again there will be formation of fruiting body. So stepwise you have to remember. What is the name of fruiting body here? The name of the fruiting body is basidiocarp. At the tip of this basidiocarp, there are so many structures which are called basidia. I am writing singular for it, basidium. Right? In this basidium, what is going to happen? In this basidium, there will be karyogamy, there will be meiosis. And it will result in the formation of four basidiospores. Right? This will be a condition. How you will remember this? Actually, the mushroom which we will consume, right, this is the fruiting body which is called basidiocarp. Right, in this mushroom, the fruiting body which you will see here, this is the basidiocarp. When you will turn the mushroom, you will see a lot of black structures here which are called gills. Right, when you will see that gill, okay, uska wall pe you will be seeing a structure like this structure is actually called basidium. On this basidium, at the tip of basidium, there will be karyogamy and meiosis. So at the tip only, we will see there will be formation of four, okay, there will be formation of four basidiospores, right? So can I say basidiospores are formed at the tip, not supported by any covering? That's why origin wise they are exogenous. Understood? They are exogenous, right? So this is what you have to remember. Everything has become as one, I feel, right? This is for basidio and this is for asco. Understood? This is the sexual reproduction which we need to study. Up same things we have written in this chart also. If you see, we have written everything here, right? Completed? Understood? Now, this is the thing which you have to remember. If they want to ask any character from any fungus, right, and they want to make the question difficult, what they will do, you know, they will give you example of that. Just say if they want to ask anything about phycomycetes, they will say that in albugo, the asexual spores are always endogenous in nature. This is true statement or false? This is true, right? Because albuco belongs to phycomycetes. Two types of asexual spore of phycomycetes, zoospore and sporangiospores. Both are formed endogenously. Correct, right? They have mentioned parasitic, right? So you have to remember, albugo is a parasite. Albugo causes one disease, that is, it will cause white rust in crucifers. It's parasitic to mustard. Okay. Albugo, mucor, and rhizopus. These three names you must remember. Albugo, mucus, mucor, rhizopus. Rhizopus का common name is bread mold. They belong to Phyco. Ascomycetes may have to remember. Baker yeast or brewer's yeast, that is Saccharomyces cerevisiae. Penicillium, Aspergillus, Claviceps, Neurospora. Okay, Neurospora used in genetic and biochemical studies. They will ask a question, which among the following fungus is extensively used for genetic and biochemical studies? So you have to remember there, it is Neurospora, right? Then commonly they are called sac fungi. Morels and truffles also belong to asco and they are edible. We can eat them, right? Then Monascus purpureus is there, that also belongs to the same, right? Remember in micro in human welfare you have studied aspergillus, what it will do, please tell me. Okay, Basidiomycetes, agaricus, common button mushroom, Puccinia nucilago, which is rust and smut. They will be causing diseases, parasitic to the plants, right? Then puffballs, bracket fungi, club fungi, their common name. And one more is there, that is called toadstool, that belongs to Basidiomycetes, that is highly poisonous, right? And three members in Deuteromycetes, that is Alternaria, Colitotrichum, and Trichoderma, they belong to this. You can remember like ACT very easily. right? Now deuteromycetes, you'll be thinking if they don't have any sexual stage reported, then where we should keep them. That is why they are kept in a separate separate class. There is no particular place to keep deuteromycetes, because we know that their asexual spore is conidiospore, which is just like ascomycetes ka asexual spore, right? So until the time we will not be knowing that their sexual stage is having ascospore or basidiospore, we will keep it in this you can say category, that is deuteromycetes. Once we will get the idea that okay, this is making a sexual spore, sorry, this is making sexual spore which kind, we will take it and we will again rearrange either in asco or in basidio, depending on which kind of sexual spore we are going to find, right? So that's why they are present in a transitional type of class for some time. Okay, once we get the knowledge about their particular sexual spore, it can be either given to ascomycetes or it can be classified to basidiomycetes. Understood, right? So this we had to remember for fungi, right? Now these are the images which are given. You must remember they have given the first image for mucor, right? Here they have given aspergillus, which belongs to which particular group, you now you can understand, ascomycetes, and this is basidiomycetes member agaricus, right? Now this has completed fungi. Now we have to study about viruses, viroids, prions, right? And lichens are also there, right? Up these are the you can say entities which cannot be classified by R.H. Whitaker. See, kingdom Plantae and Animalia is taken as a separate chapter, so I'm not including it here, right? Just one paragraph is there, you just read it and that's it. Not much is there in that. Only you have to remember they have given about alternation of generation in plants, and they have given about you can say some parasitic plants like Cuscuta and insectivorous plants, right? So except that all are autotrophic in Plantae and Animalia, so they have not given anything which you can study from that, right? No questions can be asked. Okay, next part we will go. These are certain entities which cannot be classified by R.H. Whitaker. They cannot be a part of Monera, they cannot be part of Protista, they cannot be classified as fungus, plants, animals, anywhere. So they are left unclassified, right? So we have to study them also. They are first of all not living organisms, they are acellular. They don't have anything like cell. They, viruses especially, they are obligate intracellular parasites. They need a host cell, then only they can survive, they can function, right? Then biochemically they are nucleoprotein. They are made up of only two things, a nucleic acid and a protein. The nucleic acid can be DNA or it can be RNA. Along with that, a protein should be there, right? Protein, DNA, RNA, three things together can never exist in a virus. Either it should be DNA plus protein or it should be RNA plus protein. Then the genetic material is infectious in nature. If you are thinking that which component is entering in your body which is causing infection, so you have to remember it is either DNA or RNA, or you can say genetic material. Protein cannot do anything, right? Protein is not infectious in nature. Either it is DNA or it is RNA. Now there are some historical background which is given in NCERT which you have to remember. In 1892, Dmitry Ivanovsky was the person who predicted that there is one disease called tobacco mosaic disease, where in the leaves of tobacco plant he saw certain patterns of mosaic. Mosaic means some colors, some like green, the green leaf is there, right? In that green leaf he saw that some patches are turning yellow, some are turning brown, right? So there is a pattern of mosaic which he observed on a green color leaf. He called that disease as mosaic disease. Tobacco plant may he observed, so the name is tobacco mosaic disease, right? Now he was not knowing that this particular disease is caused by which causative agent. So he predicted that maybe this causative agent is a microbe, and mostly microbe is bacteria, right? At that time bacteria only was discovered, viruses were not discovered, right? So what he predicted, he thought it is a bacteria. So he crushed the leaves of the tobacco plant, right? And then he filtered it through bacteria proof filter, so that he can filter out that bacteria if it is a causative agent, and he can know that which bacteria is causing this disease. But to his surprise, what he noticed, nothing came in that filter, right? The causative agent was found in the filtrate itself, right? So what he concluded, he concluded that the causative agent which is causing this disease, tobacco mosaic disease in the plant, is smaller than a bacteria. Understood? Okay. He was not knowing this is virus or what, but he at least understood this is not bacteria, this is something smaller than bacteria. And in cell the note of life, you have studied what is the size of bacteria. It is 0.022 to 0.2 micrometer. What is the size of bacteria? Two values you have studied 1 to 2 micrometer, and you have also studied 3 to 5 micrometer. Even if it is the 1 to 2 micrometer, virus is smaller than that or not. How in the sieves of bacteria proof filter virus can get stuck? It is not possible. Virus will come in the filtrate, right? So he understood that the causative agent of this disease is actually smaller than bacteria. Then there was one more scientist, M.W. Bijernik, in 1898. What M.W. Bijernik suggested? He has taken the fluid from infected plants, and he has transferred that fluid to healthy plants, and he observed that healthy plants will also get that infection. So he concluded that this is a poisonous fluid, right? And he said that contagium vivum fluidum, right? This is a infection causing living fluid. Understood? Then W.M. Stanley in 1938, he was the person who actually was able to crystallize viruses. He was the person who was able to crystallize virus. And because of this, we understood that it is not a living thing, right? Can you crystallize a cell? No. But maybe you can crystallize a component of cell. Maybe protein can get crystallized. Maybe the sugars can get crystallized. Maybe nucleic acid can get crystallized. But you can never crystallize a living cell, right? Crystallize means you have to convert into crystals, convert anything into crystal. So viruses, he was able to crystallize virus. And when he checked that, okay, these crystals are mostly having what, so he understood that majorly these crystals are composed of protein. Matlab protein component is more, right? This is something which you have to remember. Now generally the viruses which are going to infect plants, right, they are having genetic material which is single stranded RNA. Those viruses which can infect animals, they can have single stranded RNA as well as double stranded RNA as the genetic material. It can also have DNA, double stranded DNA as a genetic material. So all three possibilities are there. And bacterial viruses, which are also called bacteriophages, right? Bacterial viruses, which are also called bacteriophages. These bacteriophages, they have double stranded DNA most commonly as their genetic material. But if you remember, you have studied that there is one more bacteriophage that is called QB bacteriophage, which is there in molecular basis of inheritance. There you have studied having single stranded DNA as the main genetic material, right? Then what are the symptoms which viruses can cause on plants, right? So you will see mosaic patterns in the leaf, yellow, brown, right? Chlorotic, necrotic patches, right? Leaf rolling and curling, right? Abnormally rolling of leaf, abnormal curling of leaf, right? Yellowing, right? Loss of chlorophyll, vein clearing, loss of chlorophyll near to the veins, right? Dwarfing, right? Retarded, stunted growth. These are some of the symptoms which you can see on the plants, right? Four are there, so they can replace one with anything and they can ask a question to you. Viral diseases in animals. What are the diseases viruses can cause in animals? So mumps, smallpox, measles, right? Influenza, very common, right? And in humans it can cause AIDS also, right? So these are certain things about viruses which you have to remember. Now these are the two types of viruses which are explained in detail in NCERT. If you will see here, they have given this one. This is tobacco mosaic virus and this is bacteriophage. You can see here the nature of genetic material is single stranded RNA, which is surrounded by so many small protein subunit. One of this protein subunit is called what? Capsomeres. And all these capsomeres are making a protein uh, coat or a structure, covering structure, which is protective in nature for the genetic material, that is called capsid. So capsid is actually all the capsomeres present together, right? It's protecting the genetic material which is present inside. It's a helically coiled RNA, right? It causes a disease which is called tobacco mosaic disease. Here you can see bacteriophage, which is a virus which infects bacteria. Its host cell is virus. Here the host cell will be a plant, which is tobacco plant, right? Here you can see the labelings as well. This is the head part, this is a collar part, this is a sheath part. It is contractile in nature. Then you have tail fibrils, you have the end plate, right? With this tail fibrate, it will attach to the bacterial cell. Okay, this is contractile, it will contract, and the genetic material which is packaged double stranded DNA present inside the head will be delivered to the viral cell, right? So this you have to remember. Now we can compare this virus with the other acellular entities which are viroids and prions. You have to remember, biochemical nature of virus was, it is a nucleoprotein. It can have DNA plus protein or it can have RNA plus protein. But what is viroid? It is a small molecular weight ka RNA, right? A very small RNA which can cause disease, that is called viroid. Okay, a protein which is folded abnormally folded protein which can cause disease, that is called prion. So biochemical nature you understood. This is nucleic acid plus protein. This is only RNA. This is abnormal protein, abnormally folded protein, right? Here the infectious part is genetic material. Here the infectious part is also genetic material, right? Here the infectious part is a protein, right? Molecular weight mostly is coming from protein because the crystals mostly have protein. Here molecular weight will be very low because a small RNA is only there, and here the molecular weight will be somewhat similar to viruses or not, because this is just proteins only, right? So in weight these two will be similar, viruses and prions, right? Then it causes many diseases. Viruses are different types of viruses, right? You have tobacco mosaic virus, you have bacteriophages, you have COVID viruses, right? Influenza virus, many viruses are there. They causes different diseases in different hosts. Okay, viroids will cause one disease in plant, that is called potato spindle tuber disease, right? It causes a disease that is called potato spindle tuber disease. Okay, this was discovered by T.O. Diner in 1971, right? Here discovery name is not given, is Louis Pasteur, but is not given, it's removed. Here discovered by T.O. Diner in 1971. You have to remember this value. Here they have not given, they have given the name of disease. This is causing one disease actually which is a neurological disorder. Mostly it will impart the brain activities, neurological disorder. So CR Jacob disease or Creutzfeldt Jacob disease. Then mad cow disease, similar type of disease in cattle called mad cow disease, which is also neurological disorder only. Or bovine spongiform encephalopathy, BSE. This is the diseases which are caused by prions, right? Here potato spindle tuber disease, the size, or sorry, the shape of the potato will become spindle shape here, elongated type. Here there is a neurological disorder, right? In humans this is CR Jacob disease, and in the cattle it is mad cow disease or BSE. Okay, now very fastly we can do certain questions and we can move on to next chapter, that is plant kingdom, right? So see which of the following is correct statement. This is a PYQ. Okay, mycoplasma have DNA, ribosome and cell wall. Cyanobacteria group of autotrophic organisms classified under kingdom Monera. Bacteria are exclusively heterotrophic organisms, right? And slime molds are saprophytic organisms classified under kingdom Monera. Please tell me what is the correct answer. Correct answer is this one only or not? Mycoplasma have DNA, correct. They have ribosome, correct, but they don't have any cell wall. Cyanobacteria is group of autotrophic organisms which is present under kingdom Monera, correct. Monera. Bacteria are exclusively means only heterotrophic. No, bacteria is both auto, in which you have chemo auto as well as photo autotroph, remember. And if you talk about heterotroph, then bacteria is showing all categories. It's parasitic, it's saprophytic, as well as it is symbiotic also. Understood? So this is exclusively heterotrophic, wrong. What about this autotrophic, right? Slime molds are saprophytic organisms which are classified under kingdom Monera, or they are classified under kingdom Protista, right? So you can see same question which we have studied in this revision, very easy. Which of the following is incorrect about cyanobacteria? Cyanobacteria lack heterocyst. Cyanobacteria means blue-green algae. Blue-green algae will do nitrogen fixation in some specialized cells which are called heterocyst. They don't lack heterocyst. This is incorrect, right? You have to find incorrect statement. They form blooms in polluted water body, correct. They have chlorophyll A similar to green plants. They are phototrophs, correct, right? Then the primitive prokaryotes responsible for the production of biogas from, ab dekho, biogas production from the gut of ruminant. What is the answer? Methanogen. Keyword you have to focus in this chapter. right? Wrong statement you have to find. Cell wall in members of fungi and plantae. It is present or absent? It is present. Okay. Mushroom belongs to Basidiomycetes. Agaricus is the scientific name. Yes. Pseudopodia are locomotor feeding structure of sporozoans. Sporozoans, do they have any locomotory structure? No, they are non-motile, right? Pseudopodia is present in amoeboid protozoans, right? So this is wrong statement. Mitochondria are powerhouse of cell in all kingdoms except Monera. Correct. Monera does not have membrane-bound cell organelle, right? Then, which of the following organisms are known as chief producers in oceans? Please write the answer. What is the answer? Diatoms, right? Diatoms are chief producers of oceans. Ciliates differ from all other protozoans, right? Ciliates, they have flagella? No, they have contractile vacuole for removing excess water. I told you ciliates have two openings: gullet for the entry of water laden food towards inside of the cell, and contractile vacuole through which they can remove excess water, they can remove metabolic waste. Pseudopodia for capturing prey, it is there in amoeboid protozoan, having two types of nuclei, that is they are in all the protozoan, which is not in our syllabus, right? This we have not studied. Actually there are two type of nuclei in paramecium. Okay, one small nuclei, one big nuclei, macro and micronuclei, that is common to all, right? That's why this is not the answer. In which group of organisms cell wall form two thin overlapping shells fit together? You know the answer, right? Two times we have revised. That is chrysophytes or diatoms, right? Then you have to match list 1 with list 2. Tell me, for this question you must memorize the example. That's why I have given all the examples of kingdom fungi together, right? Please revise it. Rhizopus is name of common bread mold, right? It's bread mold, so it is 3 here, right? Ustilago is a smut fungus, 2. Puccinia is what? It's a rust fungus, 1. Agaricus is what? Sorry, rust fungus is 4. Here agaricus is this one. So 3142. Where is the answer? 3142. I think I have done something. Ustilago is smut fungus. Yes, it is 2 here, right? And 1 here. So 3241. 3241 is option number C. Okay. Now, which of the following statement is incorrect? Morels and truffles are edible. That is edible only. Toadstool is poisonous. Claviceps is source of many alkaloids and LSD. This is correct. It is not given in NCERT. The claviceps is actually responsible for hallucinogenic agent ki tarah se it will work, right? These are hallucinogens. They are derived from claviceps. Conidia are produced exogenously and spores endogenously. Correct. Yeast have filamentous body. Yeast. Yeast is having filamentous body, or yeast is like this, right? It is unicellular structure. How it can have filament? Unicellular means it's having single cell. Filament means it is having multiple cells, right? It's not thread like, it's not hyphae. Its body is only single cell. It's an exception to having hypha or hyphae, right? So this statement is wrong. Okay, I think one question was there also. Yes. Which of the following matches is correct? Up for solving this question, maybe every example you don't know. Phytophthora is an example of phycomycetes, but even if you don't know, we can do it. Mucor, phycomycetes, reproduction by conjugation. Okay, this we have not studied. It's a specific type of plasmogamy. Okay, here what they have given. Tell me, mucor. If you, even if you don't know, you have not studied conjugation because it's not even our syllabus. But at least you know mucor does not belong to ascomycetes, right? So this is not a correct match, right? Agaricus is a parasitic fungus, right? It's belonging to Basidiomycetes. A parasitic means it will cause any disease. What disease button mushroom will cause? If it is causing disease, we will never eat it, right? Phytophthora belongs to Basidiomycetes? No. Phytophthora is having aseptate mycelium. This is correct, but this is wrong, right? So we will not choose this. Alternaria, sexual reproduction is absent. It belongs to Deuteromycetes. Now, answer is given is D, right? But again, I will tell, it should not be written as absent. It should be written as not reported. Why I am saying this? Because we cannot claim whether it is absent or present as per NCERT line. Okay, but in such kind of question, what we have to do, we have to go with the most appropriate option. Sometimes they will not give you exact correct thing, they will give you answer as least wrong, right? So in that case we have to find this, we have to eliminate the most wrong things and we have to get to something which is least wrong. Okay. Imperfect fungi, which is decomposed of litter, help in mineral recycling, they are deuteromycetes. Remember I told you this question is already asked. Mad cow disease in cattle and CR Jacob disease in human is caused by, tell me, prion or viroid? It is prion. Viruses, they have both DNA RNA, they have DNA enclosed in the protein coat, they have prokaryotic nucleus, they have single chromosome. What's the answer? Their genetic material is packed inside a protein coat, right? Which of the following is correct about viroids? They have free RNA without protein coat, they have DNA with protein coat, they have free DNA without protein coat, and they have RNA with protein coat. So they have only low molecular weight infection causing RNA with no protein, right? So this was all about kingdom Monera. But before entering to this kingdom, I want to tell you one more thing about one thing that is called lichen, right? Actually, lichen is not an organism. Lichen is a symbiotic association between two things, algae and fungi. Okay, these two organisms grow together in, uh, you can say, uh, such an intricate relationship that they look like one organism only. That's why they are classified here, right? Sorry, they are left unclassified by R.H. Whitaker. So algae, what it will do, it's called phycobiont. This is called phycobiont because it will be doing photosynthesis, right? Autotrophic, and this one is called mycobiont. It is heterotrophic in nature, right? What will happen here? Why they are living together? Algae is providing what? Algae is providing shelter. Sorry, algae is providing food to fungus. Fungus is getting this benefit from algae, and fungus is providing shelter, moisture to algae. Right? This together is called symbiotic relationship. This is a symbiotic relationship between these two components, phycobiont and mycobiont. Uh, in this chapter, what they have written, you have to remember, lichens are sensitive to pollution. Which pollution? Air pollution. Okay, lichens are sensitive to air pollution. Basically, sulfur and nitrogen oxide के pollution से they are highly, you can say, sensitive, right? So you have to remember this. Now our next chapter is plant kingdom, right? We have to revise this chapter, plant kingdom. There are so many topics here to complete. So again this chapter starts with plant classification, right? If we consider plant classification, like biological classification, we have studied full chapter, right? Like that, initially in this chapter they have mentioned that all the plants can be categorized with the help of these three classification systems. This came early, this came later, this came next, right? So revisions happened in this classification also, right? Artificial classification system is that early classification system only, where scientists have classified different type of plants based on their external appearance or morphology, right? Aristotle is the person we have studied in the previous lecture, also divided all the plants under three categories: herbs, shrubs, and trees. Linnaeus is the person who divided all the plants on the basis of the morphology of its andrecium. जैसे कि he counted how many number of stamens are there, they are joined together, monoadelphous, polyadelphous, diadelphous, right? Or they are free. So these are what? These are the characters of andrecium which are visible to us. Based on this, he started classifying all the different type of plants under different categories. So these classification system, just consider by yourself. Only if it is based on one or two characters, right? Will it be good classification system? No. Another thing, if you consider Linnaeus work, what he did, he started giving equal weightage to Okay. Another classification you have to remember, people started classifying like herb, shrub, and tree, right? People used to classify on the basis of vegetative characters also, which are visible to us, right? So you tell me, can we give vegetative character and reproductive character as equal value? No, right? Equal weightage cannot be given to vegetative and reproductive characters. Vegetative characters, if you are considering like, okay, how many number of leaves are present, what is the size of leaf, what is the shape of leaf, what's the color of leaf, how many you can say branches are present, if we consider this kind of characters, and if we are trying to classify, or if we are just considering herb, shrub, tree also, this is also morphological character based on vegetative character, right? Then that classification system will not be good, because vegetative characters are easily influenced by environmental factors, and reproductive characters are less influenced by environmental factors. Vegetative characters are very easy to change, right? Whereas reproductive characters are more or less constant characters, or fixed or conservative type of characters. Just like, for example, if you take a plant and keep it in sunlight with lot of like everything which is needed for a plant to grow, you are providing everything naturally to the plant. Another plant you keep in some corner of your room where no sunlight is coming, adequate temperature is not there, nothing is there, right? So what will happen, you will see the plant which is kept in nice environmental conditions, it will grow very good, big leaves will be there, everything will be there perfect, right? But this plant, it will be not growing that much vegetatively, but when the time will come that it will receive the same amount of photo period, both will flower, right? So what is this? Reproductive characters are actually more important for classification as compared to vegetative character. Equal importance we cannot give, because vegetative characters are easily changed by environmental conditions, but reproductive characters are more or less conservative characters, they will not change. This is the drawback of this classification system, artificial classification system, right? Equal weightage given to vegetative and reproductive character, and very few characters considered, that is only morphological characters for classification, right? Then it was revised, another classification system came, which was called natural system of classification, right? So natural system of classification was given by George Bentham and Joseph Dalton Hooker, right? This is the most important type of classification system which you study, right? This will give you understanding about the entire overall classification of plants. So here this classification system was based on natural affinities between the characters, right? So means how closely two different plants are connected. That connection you can build only when you are reading multiple characters, right? So in natural system of classification, along with the morphological or external character, scientists have also considered more extra characters which are not visible to us, internal characters, like ultrastructure of a cell, anatomy of a cell, embryology of a cell, phytochemistry of a cell, right? All these characters are also studied, right? Which gave us a better idea about the plants, right? We can classify them more perfectly. Only one problem was there, that evolutionary relationship was not considered. So one more time revision came and phylogenetic classification system appeared, that was based on evolutionary relationship. It says that if there is any taxa which belongs to same genus, then it must have a common ancestor, right? Let's suppose we have two plants, right? Both two plants belong to same genus, one is Solanum nigrum, one is Solanum melongena, same genus, right? Taxa at same genus, Solanum genus, so they must have a common ancestor, right? In the past there must would have been a common ancestor. Because of some changes this genus appeared, because of some other changes this genus appeared. So how we are bringing the relationship between them? By finding their common ancestor. If a common ancestor is living, then we can go and check the exact plant, right? If it is dead, then we have to track the fossil records, right? So you have to remember this was given by Engler, Prentill, Takhtajan, Hutchinson. There are different scientists who have worked for phylogenetic classification system. We are actually following this kind of classification, and RH Whittaker classification system is also having this phylogenetic you can say relationship, right? Remember. Now there are certain tools which can be helping us if we are classifying different type of organisms. That includes numerical taxonomy, phytotaxonomy, and chemotaxonomy. What is the meaning of numerical taxonomy? Thousands of the characters can be studied all at once if they are fed in computers, right? So computers can be used for processing the data very quickly. right? Because it will convert your multiple characters into numerical values 0 and 1, and it can process it very fast, right? So we can use numerical taxonomy, right? Or we can use phytotaxonomy. Phytotaxonomy means studying of phytochemicals which are present in the plant. Just like if you want to build relationship between two plants, you can look at their DNA. DNA is a phytochemical, it's a biomolecule present inside the nucleus, right? So you can have a look at the phytochemistry. You can see, check for different proteins which are there, some specific proteins which are there in one plant family only, certain you can say aromatic compounds which are found in that group of plants, right? So like that, different chemicals which are there in that plant, you can search for them, right? Chemotaxonomy. Up chemotaxonomy. Okay, this word I think I have to write cytotaxonomy here. Okay, so it's not chemo, it's cytotaxonomy. Phytotaxonomy, or here we can write chemo. Okay, phyto means what are the plant chemicals which are present there. Chemotaxonomy we can consider it. Cytotaxonomy, the third one is cytotaxonomy, not chemotaxonomy. What is cytotaxonomy? Cyto actually talks about the cell, right? But we have to remember, within the cell there is a nucleus, within the nucleus there is chromosome. So if we are studying about chromosome, structure of chromosome, number of chromosome, right, which kind of division is occurring, mitosis or meiosis, behavior of chromosome, then it is called cytotaxonomy. So this all also will give us additional information about a plant, and that information we can use for classification, because as many characters, as many informative characters which we are getting for any particular organism, and we are using it for classification, our classification system will become more better, it will become more reliable, right? So these are supporting tools which we can use for plant classifications. Now we have different type of plants under plant kingdom chapter. You know, it starts from very simple type of plants, algae, then bryophyte, then pteridophyte, then gymnosperm, then angio we are not going to read in detail because it is removed from NCRT, because in the upcoming chapters only we will talk about angiosperms only, so we will limit it up to gymnosperms, right? So see, algae you know very well, it is chlorophyll bearing autotrophic plant, simple plant which can do photosynthesis, which is multicellular. Some of them are actually unicellular also, right? They all are autotrophic. They are simple type of thalloid plant. You will never see a leaf, organ or leaf or stem, anything. They are largely aquatic in nature. Most of them they will be found in water also, only like fresh water, marine water. Sometimes you will see near the stones, rocks, wherever moisture is retained, you can find algae there also. Maybe in some wood where moisture is retained, there also you can find it growing. But definitely it is not utilizing anything from that decaying wood and all. It will be doing photosynthesis because it is chlorophyll bearing plant, right? Now you can also see them on the coat cover of sloth, right? Sloth is one very lazy type of animal, right? On its coat cover it is present. Uh, it is present as associated with the fungus in lichens. Just now we have read. If you will look at the body of algae, it is having so many forms, right? Different size, different forms are there. It's highly variable. Sometimes you will see algae can be a single cell like chlamydomonas ka cell or chlorella. Sometimes you will see they are making filaments, right? Multiple cells are joining together and they are forming a multicellular filamentous structure, that is found in Eulothrix and Spirogyra. Or sometimes you will see that they are living in colonies, multiple cells are packed in a gelatinous covering and they are residing together. This is called colonial. You will find it in volvox, and they can have massive forms which can grow up to 100 feet also, that you will see in kelp, which is a marine algae, brown algae, right? So they can be colonial, filamentous, unicellular, massive, right? Their body organization is like this. Reproduction, they can show all three type of reproduction. Vegetatively they will reproduce by fragmentation. They will form asexual spore, that is a motile asexual spore in the algae, that is called zoospore. Very important, you have to remember. Sexual reproduction is of three types, okay? Isogamous, anisogamous, and oogamous. Okay, in isogamous, the gametes which are fusing, male and female gametes which are fusing during sexual reproduction, both will look exactly identical, right? Example is Eulothrix and Spirogyra. Only difference is here both the fusing gametes are motile. If it is motile, if they are flagellated and they both appear exactly same size, that you cannot predict which is male, which is female, then it is called uh, isogamous flagellated type of you can say reproduction. This is there in Eulothrix, right? You have to remember flagella is having L, Eulothrix is also having L, or motile is having L, Eulothrix sorry, motile and flagellated Eulothrix is there. Okay, flagella is present. You have to remember like this: flagella is present, so one L should be present in the spelling, right? Flagella present, L is present in Eulothrix, the spelling. So it is flagellated isogamous. Non-flagellated, two gametes are fusing, both are non-motile, right? But both are identical, that you cannot understand which is male, which is female. So it is there in Spirogyra. So the Eulothrix Spirogyra, you understood. Anisogamous, when the fusing gametes are unidentical, right? One is bigger, maybe one is smaller, right? I'm drawing here uh, motile gametes here. So in Eudorhina you have this condition, right? The fusing gametes, they are unidentical, one is maybe big, one is small, both are motile here in this case. Oogamous, oogamous reproduction, you have to remember always, and always you have to remember, the one which is female will always be very large and it will be non-motile, right? If you have a female gamete which is very large and non-motile, then it is called oogamous. The male gamete can be non-motile also. The male gamete can be motile also, right? The female gamete should always be non-motile and very large. This is there in Volvox and Fucus, a brown algae, right? So you have to remember this. They ask questions on this. This already they have asked questions in PYQ. This is very, very, very important part. You must remember all these examples. Plant kingdom is only a game of examples, right? You have to remember. Now, this is some of the important points about algae, that half of the total carbon dioxide fixation on the planet is actually done by algae itself, because most of the, you can see, algae is present in aquatic condition, right? 70% more than that of our earth's crust is surrounded by water. In that, algae is there, it is doing photosynthesis. So half of the CO2 fixed in on earth is by algae only. They are primary producers in oceans. Primary producer means they are the present at first trophic level. They are converting solar energy into chemical energy, right? First members, right, to do so. And if they are doing so in the water, definitely they will increase the amount of dissolved oxygen in water body or not, because they are doing photosynthesis, releasing oxygen. Where that oxygen will go? In water, right? So it is increasing the dissolved oxygen. There are 70 species of algae which are edible, which you have to remember. Laminaria, sargassum, porphyra is given as an example of those 70s, like some of the example from those 70 species. Then algae produce a thickening agent in their cell wall, outer cell wall, which is called hydrocolloid, right? That is of different types. That is algin. Algin is a hydrocolloid which is present in the brown algae, and carrageen and agar is a hydrocolloid which is present in the red algae, right? Agar is obtained from Gelidium and Gracilaria, and it is used for growing microbes, right? They will be used for making the growth medium, or you can say the culture medium, right? They will be used for making culture medium. जैसे कि soil is a culture medium for plant, ऐसे ही culture medium is made up of agar for seeding bacteria. Bacteria has to be kept there so that they can grow, right? So that is a jelly like thing is there, that is called culture medium, which is formed from agar, and it is also used for thickening, like as a thickening agent in ice creams and in jellies, right? Chlorella is a unicellular algae which is taken by space travelers when they are going in space. They cannot carry tons of food with them, so they will be using this supplement which is highly rich in protein. They will keep the tablets from this chlorella, and they'll take it there to space, right? Now we have divided all the algae majorly on the basis of the main pigment which is present in them, the reserve food material which is present, the type of reproduction which they are doing, right? So we have three different type of algae: chlorophyce, pheophyce, and rhodophyce, right? What's the common name of chlorophyce? Green algae. What's the common name of pheophyce? Brown. And what's the common name of rhodophyce? Red algae, right? Plant body, we have to remember what kind of plant body is present in them. So you have to remember that here you can have all three possibilities: unicellular, colonial, filamentous forms. Here in NCERT it is written there is a great variation in the size and form of this particular algae. Brown algae may some algae is there where you will see branching is there, right? So branching can be very simple branching, right? Here it is mentioned ectocarpus is an algae, brown algae, where you have simple branch filamentous forms, And kelp is a brown algae where you have high branching, right? More branching, profusely branch condition is there, right? And it can grow up to 100 meters also. This much big it is, right? Kelp can grow up to 100 meters. Massive body it is having. Simple branching is there in ectocarpus, and profusely branch, matlab more branching is there in kelp, right? Then if you consider rhodophysi, members here are multicellular. They have complex body organization. Only that keyword we have to remember, that their body organization is highly complex, right? Then which kind of pigments they carry? So here you have chlorophyll A and B. Here also you have chlorophyll A and C, and here you have chlorophyll A and D. So you have to remember, in green algae you have AB, just like higher plants. Here you have A and C in brown algae, and in red you have A and D, right? Along with that, you have the accessory pigments also studied in photosynthesis chapter. You can see here it is mentioned carotenoid and xanthophyll. And you have to remember, because it is brown, so it must have a unique pigment in it, that is fucoxanthin. Fucoxanthin is that brown pigment which gives brown color to the brown algae. R phycoerythrin is the red pigment which gives this red color to the red algae. And they have mentioned one more point, that if you will see the thallus of brown algae, you will see that color can range from olive green to various shades of brown, right? Light brown, dark brown, different types of brown are there. So this depends on the relative concentration of fucoxanthin along with these other pigments, right? Kitna fucoxanthin hai, based on that it will vary. Then we have cell wall. Cell wall is further divided into inner and outer cell wall, right? Inner cell wall in all the algae you can see it is cellulosic because it's plant, right? Outer cell wall here it is made up of pectin in green algae. It is made up of algin, hydrocolloid in brown algae, and agar and carrageen in red algae. There's a table in NCERT where they have mentioned that this brown algae is having this algin, which is a non-sulfate polyester, and red algae it is having polysulfate esters, right? So you have to remember sulfate is present here, it is absent here. Okay. Then stored food or reserve food material is definitely it is a starch and oil, right? And you will see one more structure in the green algae which is present within the chloroplast, right? Within the chloroplast there is one structure which is called pyrenoid. Pyrenoid is a structure which stores starch beside protein, right? So here in the center you can see protein is there, and surrounding that you can see a lot of starch is deposited. So this starch plus protein is called pyrenoid, which is present here in case of our green algae. Then we have laminarin and mannitol. This is a special type of reserve food material, a special sugar alcohol which is present in brown algae. And we have this Floridian starch. You have to remember its structure is similar to amylopectin and glycogen, right? They will write amylopectin and amylose. So you have to remember structure of Floridian starch is similar to amylopectin and glycogen, right? Then vegetative reproduction, if you will see, they all will reproduce by fragmentation because they carry filamentous body. If any algae is unicellular, it will reproduce by just a division of that cell, right? Then asexual spore will be done by zoospores here. Zoospores here, both of these spores are motile spore, but here it will be done by non-motile spore. You have to remember, red algae does not produce any kind of motile spore. Whether it is asexual spore, whether it is gamete, they don't produce any kind of motile structure, right? Red algae only, right? Here asexual spore is motile zoospore. You have to remember the spore is having this pear shaped condition. Same shape is there for gamete also, right? The spores are pyriform, gametes are also pyriform, pear shaped, right? If you remember here, in the table in NCERT they have mentioned that 2 to 8 flagella can be present at the tip of the cell here. So it can be a 2 to 8 equal length ka flagella, right, inserted at the apex in green algae. Here you can have two flagella, okay, only two flagella which is present on side. This is not tip, tip is this region, right? If the flagella is present laterally, if it is attached on the side, then it is called present in brown algae, right? One is small, one is longer, so it's unequal length ka flagella, right? Both are having same shape, but insertion of the flagella is different, number of the flagella is different. Here 2 to 8 apical, here only 2 lateral, here no flagella at all, right? So this is about asexual spore. Now sexual reproduction is all three types are possible in brown algae and green algae, but only oogamous type of sexual reproduction is found in case of red algae. Along with completing the reproduction after the fertilization, you have to see that several post fertilization complex stages are also found in red algae. Only you have to remember that keyword. If you talk about sexual spore, right, then sexual spore is also pear shaped with 2 to 8 flagella apically inserted. Here pear shaped, 2 flagella laterally inserted, one longer, one small shorter, just exactly same as asexual spore, and here it will be non-motile gamete, right? So this you have to remember, right? This I'm talking about, not sexual spore, sorry, this is gamete. This I'm talking about is gametes, right? They don't have sexual spore here, so just the gametes you have to remember. If you remember the name of examples, again here they will ask a lot of questions: Chlamydomonas, Volvox, Eudorhina, Spirogyra, Eulothrix, right? Cara, chlorella. And in photosynthesis chapter, remember T.W. Engelman studied filamentous green algae cladophora, so that also is written here. You have to remember all these belong to chlorophyceae. Actocarpus dictata, Laminaria, Sargassum, Fucus, and Kelp, they all belong to they all belong to brown algae Phaeophyceae. Polysiphonia, Porphyra, Gracilaria, and Gelidium, they belong to red algae Rhodophyceae, right? So you have to remember this. They have even mentioned that red algae are formed in warmer areas, right? They have also mentioned that in brown algae you will see in the center there is a vacuole and the nucleus, right? They have also mentioned that definitive chloroplast are present in green algae, right? And the shape of chloroplast can vary in green algae: disc shape, rod shape, star shape, all type of shapes are mentioned, right? So those things are also there. The gamete and the spores, asexual spores, are exactly identical, right? And then you have to remember oogamous reproduction may female gamete is always very large, non-motile. Male gamete can be motile or non-motile. In this case, in red algae, male gamete is always non-motile. Okay. Then here are the images which they have given. This is a colonial form. This is a filamentous form of green algae. Here you can see they have shown you some structures which are present in the brown algae. In brown algae, you have a photosynthetic structure which is called frond, which is just like leaf in function, but it's not it's not a multi-layered structure like leaf. It's just single layer, single line of cells is there. So frond is carrying all the pigments. It is photosynthetic. It's a stalk-like structure called stipe, right, which is like a stem, right? I am just saying like with respect to function, not structure. Hold fast is a structure by which this entire algae is holding the substratum, maybe the soil or maybe the seabed. So hold fast is there, which is responsible for just attachment purpose. Stipe is there, connection. Frond is there, photosynthetic. Then in this algae you can see they have even shown you some air bladders, right? These air bladders are providing buoyancy to the algae, right? And here you can see branching is there. Here there is only one single structure they have given. So these are certain brown algae. Even they have given the name that first is Laminaria, second is Fucus, and this is Dictata. So you have to remember their names also. What if they will give one name and they will ask what this figure is telling you, right? And then here you can see two red algae they have given. This is Porphyra, this is Polysiphonia, right? So you can see in Polysiphonia more branching is there, in Porphyra very less branching is there. Fronds also they have shown here, right? Understood. So only that much is there in algae. Please read it from NCRT. Very easy, right? Bryophyte. Now bryophyte you are going to find where? Bryophytes you are going to find in moist shaded conditions near the hills. You might have seen mats are covering the hills, cushiony mats, right? They all are small tiny plants which are called bryophytes. They are called amphibians of plant kingdom, right? Why? Because for completing their sexual reproduction, what is needed? For their sexual reproduction, water is required, right? For completing their sexual reproduction, they require water, right? So they are called amphibians of plant kingdom. Their main body is called gametophyte, right? If you remember alternation of generation, all the plants they have two stages in their life cycle: gametophyte and sporophyte. Gametophyte meaning is haploid stage. Another meaning is it is going to bear sex organs to produce gamete, right? And What is the meaning of sporophyte? Sporophyte ka meaning is it is a spore producing stage or a diploid stage. So I am saying that a bryophyte ka plant which is visible to you in green color, that is a gametophyte. Each and every cell of that gametophyte is haploid in nature. It will bear multicellular jacketed sex organs. Up to algae, the sex organs are unicellular only. Here you have multicellular jacketed sex organ. The name of the male sex organ is antheridium. The name of female sex organ is archegonium. Antheridium is a club shaped structure like this, right? And archegonium is a flask shaped structure like this. There is a egg cell, okay, which is a female gamete here, right? And in this there will be formation of this twisted antherozoid, right? Antherozoid, which is a haploid structure. Here also haploid structure, it's a male gamete, right? So it's a biflagellate twisted type of condition. If I draw here, it will look like this, right? A biflagellate twisted type of gamete is there here. So its flagella is there, that's why it will swim in water and it will transfer to the female reproductive part and fertilize there, and it will form a zygote, right? Now you have to remember, this is some important points which are given. Some mosses are actually edible, herbaceous mammals, okay, uh, and birds and other animals, they will be eating them. Sphagnum ka some species are used for packing material because of the high water holding capacity. What will happen? They will absorb lot of moisture, right? And then you can use it for you can say shipment of certain living material. Like if you want to transport one plant from this place to some other state, what you can do, you will add this sphagnum moss, right? Peat moss it is called, and it can hold lot of water, so you don't need to water for 4 5 days. It will be like going without any problem. So it will be used for transshipment. It can give us fuel in case of peat moss, right? Many mosses along with lichens, what they will do, they will be converting the rocks into soil, right? So rock decomposition they will do because of secreting certain acidic compounds. What will happen? Rocks will get like it will take millions of years, but the rhizoids which are present in mosses and the lichens, which they will release certain acidic compounds, the rocks will be broken, weathering of rock will occur, and plant succession will begin. So on bare rock, if they want to convert that bare rock into soil, these are mosses and lichens are the pioneer community, they will be growing there first, right? And they also form dense mats on the soil, so they will be helping in soil preventing soil erosion, right? Now if I talk about all the bryophytes which we have, we can divide them under two categories: liverworts and mosses. These are simple type of bryophytes, these are little bit advanced type of bryophytes. If I talk about liverworts also, we have two category. We have thalloid liverwort and we have leafy liverwort, right? What is this thalloid liverwort? Can you see the growth of this liverwort is, it will grow in prostrate condition, right? And can you see the thallus is dorsiventrally oppressed? It's appearing like somebody has sticked it on the soil, right? So it will grow like this in a prostrate direction. This will also spread only, it will never grow up, right? But this one, it grows in erect direction. Thallus will grow in erect direction, right? It is not dorsiventrally oppressed, it will grow like this straight, right? Another thing, if you see in liverworts, especially if I take, okay, liverworts, you have to remember they will have rhizoids, right? Rhizoid is a root like structure, it's not root, it's a root like structure, okay? Here the rhizoids are unicellular and they will always remain unbranched. There is no branching, there is only single cell which will help in absorption of mineral and water. Here you have to remember the rhizoids are there, but the rhizoids are multicellular and we have branching also. Okay, what is this leafy sorry, thalloid type of liverwort? In thalloid type of liverwort, you can see dorsiventrally closely oppressed type of thallus is there, right? But in leafy, can you see here the leaves are arranged in two rows of leaf like structures, okay? You can see here, this is one row and this is the another row in which leaves are arranged, right? But here, can you see any such kind of arrangement? No. In this, you have to remember, here in this case, the leaves are, or leaf like structures, leaf like structures, they are arranged spirally. All are arranged spirally, right? This is the basic difference. Understood? अब liver wort में if I take thyroid liverwort, right? I have certain example of thyroid liverwort. Leafy liverwort example is not there. One example is given in NCERT, which is Markensia, right? If I take an example of Markensia, I have to remember that Markensia is the one which produces two different thallus, right? Male thallus and female thallus. So can I say Markensia is dioecious in nature? Okay, this is dioecious plant ka example. Okay, I'll show you the image also. We have to study the life cycle of all of these things. Whatever you are looking at, this is actually a gametophyte, right? Gametophyte is thyroid or leafy. Gametophyte is in this is of this kind, right? Sporophyte generation we have to study. There are two type of generation. One is gametophyte, which will be bearing sex organs for gamete formation. After that, there is a stage which is called sporophyte. I'll show you that. Okay, Markensia, you have to remember it is dioecious. Okay, I'll show you Markensia ka image. See, this is dioecious. Can you see Markensia is bearing this archegoniophore, on which archegonium will be attached on this disc. And here, can you see this is anthridiophore, a stalk-like structure on which anthridium will be present on this disc, right? So these two different thallus are present, male thallus, female thallus in case of Markensia. Whereas you can see this is how a moss will appear. Okay, this is how a particular gametophyte of moss will appear. You can see here there is antheridial branch, right? There is archegonial branch, two different branches, right? After the fertilization, you will see one structure like this where gametophyte and sporophyte is present together. So I'll show you this later. First, I'll explain you about the life cycle of liverworts. So first thing you have to remember, if we are talking about a liverwort, let's suppose here I have mentioned this liverwort is let's suppose one thallus I have drawn, and in that I am showing you both sex organs are present, male and female sex organs, right? So let's suppose here you have that receptacle, right, where a disc is present, where you have antheridium, and here we have this stalk where this disc is present, and here we have this archegonium. The name of this stalk is anthradiophore. The name of this stalk is archegoniophore. Up when we will check this disc, we will get to see lot of anthridium will be present, and here we will have archegonium, right? This is the male sex organ, this is the female sex organ. This is formed on the main plant body, which is a gametophyte. Gametophyte is green in color. Gametophyte is multicellular. Gametophyte is haploid in nature. So if I want to make the sex organs in this gametophyte, it will be formed by which division? Mitosis, right? Now, the work of this sex organ is, I told you, gametophyte ka meaning is it is there to make sex organs so that gametes are formed, right? This particular plant, this liverwort, it is bearing this sex organ, antheridium and archegonium. Antheridium may male sex organ, that is a biflagellated structure called antherozoid will be formed. Within the archegonium, egg cell will be formed in that venter part, right, swollen part, right? This egg cell and antherozoid, right, they have to fuse. For fusion, the antherozoid has to swim and reach to the female reproductive part, that is archegonium, within which the egg is located, right? So for this transfer, it requires water. Understood? This is the necessity why the bryophytes are not able to completely move to land, because they need water for their sexual reproduction, right? Antherozoid has to swim around and reach to the flask shaped structure, this flask shaped structure, okay, in which this egg cell is present. It has to reach here. Uske liye it requires water. Once the antherozoid will reach within this flask shaped structure and it will fuse with the egg cell, it will form zygote. Zygote will be two end diploid, right? This is haploid, this is haploid. Fertilization, syngamy will occur. zygote will be formed. Zygote will divide by a mitosis to form embryo. This is the first time the plants have formed embryo. In algae, there is no embryo, right? So this is the first type of embryophytic plant. Now this embryo, it has to form a spore, but directly it will not form spore. It will form an intermediate stage. This embryo will again divide by mitosis to form a multicellular non-green structure that is called sporophyte, right? This multicellular non-green structure, which is called sporophyte, is developed from this embryo only, right? By mitosis. Now, this multicellular non-green structure is divided into three parts: capsule, seta, and foot, right? Capsule, seta, and foot. If I begin from foot, this foot connects this entire sporophyte to the gametophyte, right? This is the gametophyte or not? This is the sporophyte. To this gametophyte, sporophyte is connected with the help of foot, right? At the tip, it is having capsule. Within the capsule, there are certain cells which are called spore mother cells. What is the ploidy of foot seta capsule? They are part of sporophyte. Sporophyte is formed after embryo formation. So sporophyte is diploid in nature. It is having no green pigment. It is non-green. So can we say it is a partial parasitic? Okay, it's partially parasitic to the gametophyte. It's partially parasitic on gametophyte. Okay, it is attached to the gametophyte. Can you see through the foot? It is attached to the gametophyte. Gametophyte is actually green in color, so gametophyte will do photosynthesis. It is photosynthetic structure, right? But this is parasitic because it is non-green, right? So it will take the nourishment from the gametophyte and it will grow. And why it is growing? Because inside the capsule it is having spore mother cells. In the spore mother cells, meiosis is going to take place and it will give rise to haploid spores. When this haploid spores are germinated, they will again give rise to this gametophyte, which is haploid in nature, right? So can you understand this life cycle? It is very easy. It is having two different phases. One is gametophytic, one is sporophytic. Gametophytic phase will be making gametes by bearing sex organs. Gametes will fuse. After fusion, they will form zygote. Zygote is diploid. It will form embryo. First plants to make embryo. Embryo will not directly make spore. First embryo will make sporophyte. Sporophyte is having no green color. It is acting like a partial parasitic structure on the gametophyte. It will drive the nourishment from the gametophyte. It is having no green color. In the capsule area, it's having spore mother cells. The spore mother cells are divided by meiosis to form haploid spores. Whenever this haploid spore will germinate, they will give rise to new gametophyte. This is about the sexual reproduction life cycle, right? Now let's suppose if they don't want to do sexual reproduction, then there are certain structures like this, right? These structures are actually called what? These structures are called gemma cups. Okay, what is gemma cup? It's a small receptacle in which we have some asexual multicellular green color buds, right? Whenever they will fall, they will produce new thallus, right? So what is this? This is a type of asexual reproduction. Understood? Asexually also they will reproduce, liverworts will reproduce. How? By forming these small receptacles which are called gemma cups. On this gemma cup, this asexual bud is present, which is called, this asexual bud is present, which is called gemma, right? This gemma is multicellular. This is not unicellular structure. It's having multiple cells like this, right? When it will get detached, it will fall down, and from this notch, it will produce two thallus, right? So this is one thallus, this is two thallus, right? These are two new thalli, right? So can I say asexual reproduction liverworts occur by formation of gemma cups, right, by formation of this asexual buds which are called gemma, right, and sexually it is reproducing like this. Okay, now let's come to mosses. Mosses also follow the strict same pattern, right? They will also make gametes, gametes will fuse, they will also form embryo, right? But there are certain differences here. What are the differences? If you consider mosses, here you can see in mosses the gametophyte is actually divided, or you can say it is having a specialized name which is called protonema. The gametophyte of moss is called protonema. This is the name written, right? If you consider this protonema, you can see I have drawn protonema like this. It's a creeping green filamentous structure, right? What I said, creeping green filamentous structure, right, which is multicellular rhizoid containing structure, right, branch rhizoid containing structure. On this primary protonema, which is the gametophyte here, right, this is the name of the gametophyte. On this primary protonema, there will be a branching which will be formed, that is called secondary protonema, right, that will grow erect. On this primary protonema, which is creeping structure, a secondary protonema will be formed which will grow erect. Secondary protonema will bear certain lateral buds. Okay, this pink color structure which we have drawn is lateral bud. Secondary protonema will bear lateral buds. Lateral buds will grow into leafy stage, right? Lateral buds will grow into leafy stage. At the tip of the leafy stage, sex organs are born, right? So this step you have to remember. First you have creeping structure, that is called primary protonema, that will form a secondary protonema as its branches. Secondary protonema will bear lateral buds. Lateral buds will grow into leafy shoot or leafy stage. Leafy stage ke tip pe you will get sex organs. There will be a separate branch for male sex organ and separate branch for female sex organ, right? So you have antheridia in the male branch, archegonia in the female branch, right? Anthrozoid, antheridia will produce, egg cell, archegonia will produce. Again same thing will happen, zygote will be formed, embryo will be formed, sporophyte will be formed on this structure which is having puncta and capsule. Within the capsule, spore mother cells will divide by meiosis to give rise to spore. Whenever the spore germinates, again a protonema is formed. So everything here is same, only this is different, right? Because the structure of the bryophyte is different. Are you able to understand? And in NCRT, they have written that the gametophyte in mosses is having two stages. Okay, if you have gametophyte to study in mosses, you have to remember. Okay, in mosses, gametophyte is divided as protonema and leafy stage. Understood? They have given like this. Okay, because leafy stage and this entire protonema is counted as one in NCRT. Okay, leafy stage and protonema, these are the two different stages of gametophyte. They have given like this, right? You have to remember, sometimes they will give question. Okay, here they have shown you mosses में two different branches, and here they are showing you a moss with a sporophyte attached on a gametophyte, deriving all the nourishment from here, parasitic, right? Okay, understood? Now teratophytes. Teratophytes mein what we have to remember, it's the first true terrestrial plant, right? It's the first true terrestrial plant because it has developed vascular tissues for the first time, right? And whenever you have a vascular supply within any tissue, that particular tissue will be bearing, like will be making true organ. Remember, if you cut angiosperm ka structure, root, stem or leaf, anything, you will be having vascular tissues like xylem, phloem there. So if you will cut teratophytes also, these are the first plants in which xylem and phloem was formed, right? So here also you will get the xylem and phloem. That's why you are saying they have true organs. True leaf is there, true stem is there, true root is there, but that is not there in the previous ones, right? Previous ones like up to bryophyte, you don't have any vascular bundle. They are atracheophytes. That's why they don't have any true organ. That's why body is having thyroid condition, right? Now the main plant body here is sporophyte. The main plant body in the previous case was gametophyte. Here it is sporophyte. They will also grow in moisture laden places like in shady places, sandy soil also some of them can grow, but yes, mostly cool, damp, moist, shady places where they will grow. Okay, their leaves can be of two types based on their size. Very, very small leaves, they are called microphyls. Like in Slagenella, it is present. and bigger leaves, they are called macrophyls, they will be present in ferns, right? A certain special type of mature leaves, when you will turn around that leaf towards the ventral side, at the margins you can get some small small structures which are called sporangia. These are the dark brown color structures which are formed only at the margin of ventral side of a pteridophyte leaf, right? In a mature leaf, that is called sporangia. These sporangia are having spore mother cells in which meiosis will take place, right? And they will be forming haploid spore. So what is the purpose of what is the purpose of sporangia? Sporangia is a diploid stage. Sorry, what's the purpose of sporophyte? Sporophyte is a diploid stage of plant which will make spore, right? So sporophyte will bear sporophyll on which sporangia will be present, within which spore mother cells are present, which will do meiosis to form spores, right? Now sometimes you will see these sporophylls are organized into a structure, compactly arranged, right? This compact arrangement of sporophylls will give rise to a structure called cone or strobilus, right? This cone or strobilus is found in Selaginella and Equisetum. You can see here all the sporophylls at the tip, they have clubbed together, right, as cone or strobilus. And here also you can see cone or strobilus formed in Equisetum. So you have to remember, cone is found in Equisetum and Selaginella, right? Then spores, some pteridophytes can be those kind of pteridophytes where two type of spores are produced. They are called homosporous pteridophytes. And some pteridophytes are produced where there are two type of spores produced. One is smaller, one is bigger. So it is called microspore and megaspore. They are called heterosporous type of pteridophytes. Selaginella and Salvinia, these are the examples of heterosporous pteridophyte which you have to remember, right? Now this is another thing which you have to remember. The name of the gametophyte stage of the pteridophyte is prothallus. Protonema is the name of the stage, gametophytic stage of mosses, and prothallus is the name of gametophyte of pteridophyte, right? It's a gametophyte. It is also green in color. It is photosynthetic, independent, but it is very tiny. It's very small. It's not even visible. It is very reduced. It will bear sex organs, which is Antheridia and Archegonia, because it's gametophyte. It is its work. It requires cool, damp, shady places. That's why the distribution is not everywhere. Pteridophytes you will get to see in very very few places where the moisture is retained only there, right? And there is one more thing which you have to remember, right? After the sexual reproduction, in some of the pteridophytes which are heterosporous pteridophytes, you can say this was actually one precursor event to form seed. So heterospore is considered as precursor for seed habit. I will tell you this after completing the life cycle. This is the life cycle of homosporous pteridophyte, right? Which is very very simple. In homosporous pteridophyte, you have to remember, they are going to produce only one kind of spore, that is homo, like a single spore. So this, let's suppose this is the main plant where you can see true roots are there, true leaves are there, stem is there, right? If I cut this particular plant, right, sorry, if I take out one of the mature leaf of this particular plant, that particular plant, that leaf will be called as sporophyll. On the margin of the sporophyll, sporangia will be there. Sporangia will be bearing diploid cells which are called spore mother cells. These spore mother cells will divide by the process of meiosis, right? And they will be forming haploid spores. When this haploid spore will germinate in the soil, it will form prothallus, right? That multicellular reduced green color structure which is photosynthetic in nature, bearing sex organs, right? So one spore has germinated, only one prothallus will be formed. Prothallus is heart shape in pteridophytes, right? So this one heart shape prothallus is formed here, and it will bear both male and female sex organs. If on one gametophyte, one prothallus, both male and female sex organs are present, can I say this is a monoecious type of prothallus? Homosporous pteridophytes always produce monoecious gametophyte, both male and female present on same prothallus. Antheridium will be making anthrozoid, Archegonium will be having egg cell. Anthrozoid will swim in water, it will reach to the egg cell, fusion will take place, zygote will develop, zygote will divide by mitosis to form embryo, and embryo will again grow into a complete pteridophyte. Only problem is, for survival of this structure you need moisture, right? So this is Very simple type of life cycle which is there in pteridophyte. If you're not able to understand it, please rewind it and see it again. We have less time and still we have two more chapters, right? So here you can see this is the life cycle of heterosporous pteridophyte. Here what is the condition? See, this is the sporophyte. Again here you have sporophylls, right? But this time I'm saying Slagenella was having cone, right? So male cone will be made up of microsporophyll or male sporophyll, female cone which will be made up of macrosporophyll or megasporophyll, right? One single leaf from that cone will be bearing microsporangium. Here also each and every leaf, one single leaf, if we will take out macrosporophyll, it will be bearing megasporangium. Meiosis will take place in this microsporangium and megasporangium to form microspore and megaspore. When microspore will germinate, it will form male prothallus. When megaspore will germinate, it will form female prothallus. So can I say here it is not monoecious, here it is dioecious type of condition which we have to remember, right? In the previous case we have seen one spore will generate only one prothallus bearing male female sex organ in one prothallus. Here two different spores are germinating, so two different prothallus will be formed, male prothallus and female prothallus. Male prothallus will bear antheridia, female prothallus will bear archegonia. This will form anthrozoid, this will form egg cell, right? Up, what you have to remember, the entire thing which I have written here is occurring within the microsporangium only. Usually what will happen, gametophyte or prothallus will be formed outside the sporophyte in the soil, right? Spores are formed, they are liberated in the soil, prothallus is formed. This is what happens in homosporous. In heterosporous, what is happening? Everything is happening within the microsporangia and megasporangia. This is microsporophyll, this is microsporangia, this is megasporophyll, this is megasporangia. Within the microsporangium, can you see now what is there? Anthrozoids are ready. Within the within the megasporangium, what is there? Archegonia is there, okay, within which egg cell is there. This is occurring on the sporophyte only. This is a cone structure, this is present on this part, right? This is happening on the main plant. Are you able to understand? This is not happening in the soil. This is the first difference. Second, once this microsporangium will open, this anthrozoids will swim in thin film of water and they will reach to the megasporangium, right? They will enter in the megasporangium, they will fuse here, they will form zygote and embryo will be formed within the megasporangium here, right? Let's suppose embryo is formed within this megasporangium. What is another name of megasporangium in angiosperm? Remember, megasporangium is also called ovule. Ovule is the thing which is going to make seed later, right? So same thing, you can analyze its kind of happening here also. Let's suppose this is a megasporophyll. Embryo is now formed here. Megasporangium, within it, it is present, right? And this entire thing is present on the main sporophytic plant, right? If I say this is a part of a sporophyte, this is a plant. So is it looking like a precursor to seed habit? This would have never happened if we don't have two different type of spores, right? Understood? But this plant actually has taken steps to make seed, but it failed. Why? Because this embryo can be retained for a longer duration within this structure, right? Even the female gametophyte, right? That is the within this megasporangium, there is a female gametophyte also in which this embryo is formed, right? This is not retained on the main plant for a permanent duration, right? So what you will have to understand here, for seed formation some steps have to be taken, then only a plant can successfully make a seed. First is heterospory condition, two spores should be there, right? Pteridophytes have successfully made it, right? They have taken at least an early step to make seed by making the two spores, type of spores. Gymnosperm, angiosperm also, always you will see there are two type of spore, micro and megaspore, right? Gametophyte should be retained on the sporophyte. This also they have done, but not permanently. Okay, after some time that particular gametophyte will fall down. Here it is permanent, right? Seed coat they were not able to make because their megasporangia was not having any integument, right? Then retention of this embryo on the sporophyte until the seed develops, this also they have failed. But can you see, gymno and angiosperms were able to do this. That's why they made seed, and they this was only one precursor step, right? They have just taken the precursor step for seed habit, that is heterospory, right? Rest all they were not able to do completely. That's why they failed to make seed, but at least they have taken a measure to make a seed, right? Understood? Okay, these are the that images which you have to remember. They have given Flagella, equisetum, fern, and salvinia, right? Up, see, there are certain things which you have to do on your part. Please remember all the examples of bryophyte. I have not mentioned sphagnum, funaria, and you can say Markantia, Rexia. All these names are given in NCERT. Please write it somewhere, okay, and remember it. Now the last type of plants, which are gymnosperms, right? Gymnosperms are the first two plants which are making seed. They are first seed bearing plants, right? Now these are vascular plants. They have secondary growth. They are terrestrial in nature. They are present in tropics, temperate mountains, almost every place where these conditions are there. Tallest tree which is given in NCERT is Sequoia or giant redwood tree, and there is one smallest gymnosperm which is called Zamia, right? Gymnosperm ka body is also sporophytic. You have to remember, if you want to do questions in plant kingdom, always remember the main plant is of which kind. Bryophyte mein it is gametophytic, right? Pteridophyte it is sporophytic. Gymnosperm sporophytic, right? Gymnosperms are heterosporous. All the gymnosperms are heterosporous. Can I write the same statement for pteridophytes? No, all the pteridophytes are not heterosporous. Few pteridophytes are heterosporous like Flagella and Salvinia. Gametophyte here is highly reduced and very few celled. Ab dekho, as we are moving from pteridophytes to gymnosperm, can you see gametophyte is even way smaller. Pollen grain is the male gametophyte, only 2 or 3 celled, right? And female gametophyte is also very few celled. They are non-green and they are completely dependent on sporophyte. If I remove this gametophyte from the sporophyte, the gametophyte will not have its independent existence. But in case of pteridophyte, at least the gametophyte was having few cells, but it was photosynthetic, so it can exist by itself. But here the gametophyte is not even photosynthetic, so it cannot exist by itself. Now this is the thing which you have to remember. They have explained two different type of gymnosperms, Cycas and Pinus, right? They have mentioned that roots in Cycas is taproot, Pinus also taproot, but here roots are associated with nitrogen fixing blue green algae, so this specific association makes a root called coralide root. Here roots are associated with fungus, so they make a specific association called mycorrhiza. Here the stems are unbranched, here the stem is branched. Leaves here are pinnately compound type of leaves, here leaves are simple type of leaves. Leaves are persistent for many years, right? Leaf will persist for many years in cycas, right? The leaves which are formed once will be there for many months, many years, right? So it's persistent for years, and leaf is covered by thick cuticle. Here leaf can withstand extreme temperature, extreme humidity and wind because of certain adaptations, right? In extreme wind, humidity and you can say temperature, leaf are going to you can say exist because they are having this adaptation. For reducing the transpiration, they have needle like leaf, they have sunken stomata, right? They have a waxy thick cuticle, right? And they are also yes, because of this they will be reducing transpiration, right? You can see this is the compound leaf, right? Compound leaf will be studying in next class, right? So this dissected type of leaf where the dissection like incision is up to the margin, this type of leaf is compound leaf, whereas this leaf is a simple leaf, it's a needle like leaf like this, right? So this is a simple leaf which is there in Pinus, this is a compound leaf which is there in the Cycas. You can see crown of leaf is present from the main stem, right? And here you can see so many branches are there in this plant, right? Like this, okay? Then what about the cones? Cone is male cone, female cone, both are present in Pinus, but only male cone is present in Cycas, female cone is absent. Only megasporophyll is there. Megasporophyll does not organize to form female cone in Cycas, right? So you have to remember it as it is, that both male and female cones are found in Pinus, only male cone in Cycas, female cone absent. Instead of that, only megasporophyll. Right? Here you have male and female plant, two different plant on two different like two different reproductive structures on two different plants. So male cone is present on one plant, here megasporophyll is present on another plant. So you have dioecious condition, but male and female both cones on the same plant, this is called monoecious condition. Right? This image I have added here, but this does not belong to Cycas or Pinus. Right? Here this is an image of ginkgo, and they can ask you what is the structure showing there, like a tomato, but you have to remember it is not a fruit, it's a seed. Right? So you have a long shoot, you have dwarf shoot, and you have a seed here. This is ginkgo, a living fossil. Okay. Now here I have given, I don't know is it visible to you or not, I think you can zoom it and you can see this. I have given a life cycle of gymnosperm. I will explain it to you. You just please listen to me. This is a pinus, so it is having both male and female cone on same plant. Right? Up, if it is pinus, if it is gymnosperm, what is the main plant body? It is diploid in nature. Right? So it should be a sporo. It should it's called sporophyte. Right? So its work is it should make spore. For making spore, it is having a male cone and a female cone. Male cone is also called male strobilus or lax, male lax, and female cone is also called female strobilus or female lax. Right? This cone is called microsporangiate. This cone is called megasporangiate because it will be bearing microsporangia and megasporangia. Understood? This male cone will be formed by which sporophyll? Microsporophyll. Female cone will be formed by which is sporophyll? Megasporophyll. Right? They should bear what? Microsporangia. Right? Megasporophyll should bear what? Megasporangia. Right? Microsporangia will have microspore mother cells and megasporangia will have megaspore mother cells. Right? In the mother cells, what we are going to do? Meiosis. So up to here everything is diploid or not? Cones are diploid. Micro and megasporophyll is diploid. Micro and megasporangia is diploid. Right? And then the spores which are formed, uh, they will be haploid because they are formed by meiosis. Now microspores are going to develop into male gametophyte, which is called pollen grain. For the first time pollen grain will be formed in any plant, then it is gymnosperm. Right? So microspores are going to develop or mature into male gametophyte, that is called pollen grain, and the megaspore is going to develop into a female gametophyte. Right? Megasporophyll is going to develop into female gametophyte. Here you have to remember, within the megasporangium there are so many cells which are called nucellus cell. Right? If I draw it here, let's suppose this is a megasporangium. Right? In this, all the cells which are present here, they are called nucellus cell. Right? In this nucellus cell, there will be certain cells which will act like megaspore mother cell. That megaspore mother cell will divide, and that megaspore mother cell, I'm writing megaspore mother cell, it will divide by meiosis and it will form one megaspore which is functional and three megaspores which will degenerate. Right? These three megaspores are going to degenerate. This remains functional. This megaspore is going to develop into female gametophyte. So in this structure, you will be seeing a female gametophyte developed here. This female gametophyte will bear archegonia. Right? How many archegonia will be formed here? Two or more than two. It will never be less than two. Right? This is fixed. You have to remember. In this, two or more archegonia will be formed. Right? So here in this structure, two or more archegonia, two archegonia we have formed here. Right? A pollen grain is ready. Pollen grain will come here and it will get deposited here. It will form the pollen tube. It will reach to the archegonium and it will release the male gamete. Now egg cell will fuse with the male gamete. What will happen once the egg cell is fusing with the male gamete? Right? Then embryo will be formed. As soon as in this area embryo is formed, these integuments will start hardening into seed coat, and outside you will see this entire megasporangium will convert into a seed. So within it, after fertilization, embryo is formed. After, like just at that time, the integuments are hardening into the seed coat, and this megasporangium or ovule is going to convert into seed. This is how seeds are formed. Because megasporangium is just there on the megasporophyll, there is no ovary present here. That's why these fruits will, sorry, these plants will never produce any fruit. They will only produce seed, right? So pollen grain is very important here. Pollination by air is important here. The male and female gametophytes are completely dependent on the sporophyte, highly reduced, right? Nucellus may one cell will remain alive. Sorry, nucellus cells will act like megaspore mother cell. One cell will remain alive after division, that is the functional megaspore that will give rise to female gametophyte, right? Pollen grains are also formed. They will go and reach there. Two or more archegonium only created or formed. That all you have to remember, right? Now this chapter we have to do certain questions. Which one of the following shows isogamy with non-flagellated gametes? Tell me. Non-flagellated gametes, I told you, if L is there in the spelling, flagella is there. So Eulothrix is not that one. We have Spirogyra, right? Spirogyra and Eulothrix are two filamentous algae which they have given example. So non-flagellated isogamous Spirogyra, flagellated motile isogamous, it is Eulothrix, right? Which of the following algae contains mannitol as reserve food material? So mannitol as reserve food material is present in brown algae, that is Ectocarpus, right? Then plant body of the brown algae is attached to the substratum by hold fast, right? Stipe is the stalk-like structure and frond is photosynthetic. Members of Phaeophyceae vary in their color from olive green to various shade of brown. Sorry, various shades of brown. It depends upon the amount of xanthophyll fucoxanthin present in the Phaeophyceae. Is this correct? Yes, this is correct. Both assertion reason are correct, and the reason is the correct explanation of assertion, right? Because how much fucoxanthin along with that yellow pigment is present in the cell, that will decide what color the cell will get, right? Which of the following algae produces carrageen? So carrageen is produced by red algae. Then the first stage of gametophyte in the life cycle of moss is protonema. In moss it is protonema only, correct? Protonema develops directly from spores produced in the capsule. This is also correct, right? The life cycle begins from here. Spore germination will result in the protonema formation. So both assertion reason are true, and the reason is correct explanation of the given assertion. Select an incorrect statement about gemma in bryophyte, right? So gemma are what? They are green, multicellular, and they are asexual buds, not sexual. They develop in small receptacles called gemma cups located on thallus. This is correct. Gemma become detached from parent body, germinate to form new individuals. This is also correct, right? Gemma are green, multicellular, asexual. So we got the answer. We have to find the incorrect one. Okay. Now, which of the following statement is incorrect about gymnosperm? Male and female gametophytes are free living. No, right? That's absolutely wrong. I told you male and female gametophyte are completely dependent on sporophyte. They are non-green in color, so they don't have independent existence, right? Rest everything is fine. Female gametophyte of gymnosperm, it bear two or more archegonia. Yes. It is retained within the megasporangium. Yes. Is it a unicellular structure? No, it is multicellular. Few cells, but definitely multiple cells. Right? Which of the following is correct for pteridophytes showing the events precursor to seed habit? Right? Non-vascular, no. Male gametophyte in these plants are retained on parent sporophyte for variable period. Development of zygote into young embryo take place within the female gametophyte. देखो, seed is formed within the female structure, right? Nothing to do with the male, right? So we have to remember that zygote is changing into embryo within the megasporangium, which is there, like within the female gametophyte, which is there in within the megasporangium, which is present on the main plant, right? So we have to choose this. That is why seed is formed on the main plant. Within the seed there is embryo, right? Try to imagine the structure of this same thing before fertilization. Megasporangium is there, within which embryo sac is there, within which egg cell is there. After fertilization things will change, right? But everything has to return on the main plant. Strobili or cones are found in Equisetum and Slagenella, right? You can see they have asked this in recently only, right? So this completes our two chapters, plant kingdom and biological classification. Now we have a next chapter, that is morphology. Please give me 1 minute. I'll just have a sip of water and we'll continue it. Okay. So now we have to resume our discussion on revising this chapter, right? Morphology of flowering plants. This is very easy chapter, but again, like plant kingdom chapter, a lot of examples are there which you have to memorize. So please revise this chapter. Please revise plant kingdom, biological classification, and morphology examples very nicely, right? So see what they are describing here. Okay, before entering into the topic, what we can do, I can tell you, like let's suppose if we are thinking about a plant, right? So plant may, this is the condition, entire plant ka body is planned on one imaginary axis, right? This is called plant ka axis, or I'll just make it little bit thinner, right? This is that plant axis, imaginary axis, right? Let's suppose this much part is under the soil and this much part of axis is above the soil, right? So this part of axis will grow in this direction, this part of axis will grow in this direction. You will call this axis growing up as ascending axis, and this one which is growing down, you will call it as descending axis. So the descending axis, can you say this is going to be root, and the ascending axis, can you say it is going to be the shoot? Are you able to understand? Right? So you have to remember the descending axis of the plant is actually the root. It is formed by radicle, right? When the seed is there in which the embryo is present, right? This part extends, right? This radicle only is going to give rise to root, right? So radicle extends and it will form root. Okay, there are different type of root systems which we have to remember: tap root system, adventitious root system, and fibrous root system. What is tap root system? You have to remember the examples. In mustard you have tap root system, in wheat monocots mostly you have fibrous root system, and in banyan tree, monstera, stilt root, you have all these as adventitious root. Just remember, if any root is formed from direct elongation of radicle, right, then it is called tap root. Okay, if I draw one figure here, you will be able to understand it more clearly. If I'm saying that this is a seed, right, within the seed, you know, embryo is there, right? So let's suppose this is the embryo, this is a plumal part, and I'm saying that this radical is elongating, right? So this root will be considered as primary root. This root will be considered as primary root, right? Then if any branch will come on this root like this, Let's suppose abhi isme to we cannot draw it, it will look abnormal only, right? Because it appears wrong. Let's suppose after some time this primary root will become persistent and little bit thick like this. Okay, this is primary. On this primary any branch will come secondary. On this secondary any branch will come tertiary, right? All these root, this primary root plus its branches together, this forms tap root system. Okay, primary root along with its branches, it is forming taproot system. You are going to find this in mustard, right? Like that you have to remember. If one more thing, if the primary root becomes persistent, then only you will see this condition. Okay, let's suppose the primary root is non-persistent. Okay, first the primary root is formed like this, and then I'm saying the primary root is short-lived, so it died, right? Primary root has been formed, it is short-lived, right? So what will happen? There will be formation of small thin filamentous roots which are called fibrous root, right? This fibrous root is formed from the base of stem. If any root is getting formed from the base of stem, then that root is called fibrous root, right? Now if there is, let's suppose this is one plant, it's having a branch, right? This is having nodes, and here also nodes are there. This is a branch, right? And if any root is formed from this branch like this, oh my god, I've drawn on the upper side. Oh, okay, if any root is getting formed from the node of the branch, right, then this kind of root is called adventitious root. Okay, adventitious root. What is the definition of adventitious root? Any root which is formed from any other part of plant other than radicle. You have class here. Can you ask that cabin? Okay, so here this is called adventitious root. Okay, now here we have given the same thing, that here we have banyan tree, may we have adventitious root, we have rice, wheat, fibrous root, we have mustard, taproot example, right? Now functions, what are the functions? Anchorage of plant, right? Roots will be providing as anchorage to the plant. It will be responsible for absorption of water and minerals, for synthesis of different type of plant hormones, and for storing the reserve food material, right? Because see, roots sometimes you have to remember that these are having cells which are non-photosynthetic in nature, so they will be storing the reserve food material, right? So you have to remember here. Now there is the next topic, that is called regions of root. In this regions of root you have to remember we have different regions. Here we have region of meristematic activity. Okay, we have a region of elongation, we have region of maturity, sorry, there's one more region here. Okay, root cap, right? Region of meristematic activity, region of elongation, and region of maturation. In plant growth and development also I have explained all these things to you, right? The first region in the root you have to remember is a protective region, right? This is also called calyptra. It's a thimble like structure which covers the entire area, where very thin walled cells are there, which are meristematic cells, right? So cells are continuously dividing over here, and they are protected by this covering, which is the thimble like covering, which is called you can say uh, root apex, right? So that root apex is always protected by this root cap, right? Now region of meristematic activity may we have cells which are very thin wall cellulose containing cell wall wala cell, then cytoplasm is there, they're actively dividing. After the division, the divided cells are moved to upper direction, where the cells are going to elongate. So here region of elongation, here the cells will elongate, right? Then elongated cells will go up, where the cells are going to mature. Here the cells will get their own roles, right? They will, some of the epidermal cells will be extended to form root hairs, the internal layers of the root, pericycle, endodermis, everything will develop in this layer only. So you have to remember, we have four different areas: root cap, region of meristematic activity, region of elongation, and region of maturation, right? Now we have different type of modifications of root which was there in NCERT. Now it is not there. So you have to remember, roots ka main function is anchorage, PGR synthesis, storage, right? And you can say absorption of water. That's a main function. Other than that, if they are performing any other function, then it is a modified structure of that root, which is because of which they are able to do it. Like in turnip and carrot, right? A tap root is modified to store food, right? In sweet potato and asparagus, the adventitious root is modified to you can say store food. Prop roots in banyan tree, have you seen hanging roots which are coming from the big branches of banyan tree, right? They are called prop roots. They are actually adventitious roots because they are formed from any other part of plant other than radicle, right? Stilt roots in sugarcane, you will see sugarcane is very thin stem containing plant. It's having so many different nodes, right? If from the basal node, if any root is coming like this and anchoring extra to the soil, providing more support for this thin long stem, then this particular root is called uh, you can say stilt root, that is there in sugarcane. Then for respiration, you have to remember, sometimes what happens that tap roots, they are coming out from the water, right? If mangroves, if you consider, mangroves are always present in areas where water is saturated, right? It's saturated with water, right? So lot of water is there, marshy condition is there, swampy condition is there, and the tap roots, they will branch up, they will grow vertically up for gathering oxygen, because conditions in that water will like in that soil will become anaerobic. They will be having devoid of they will be devoid of oxygen. So for getting more oxygen, what happens, this tap root will vertically grow up and it will come out. It is having lot of pores, right, through which oxygen can enter. So they are called respiratory roots. This is there in pneumatophores, right? It's a modification of tap root. Whereas in stilt root, you can see this is the root formed from the basal nodes, right? So this is adventitious root. Banyan tree may also, the roots are getting developed from the nodes of branch, so it is also adventitious root, right? Then nitrogen fixation के लिए sometimes in the tap root you will see some nodules will develop in which bacteria will be growing for nitrogen fixation. So that is also a modification of tap root. So they can ask these questions to you, right? Now the ascending part of the plant axis, that is called stem. This is formed from plumule. It is divided into node and internode, which is absent in root, right? The young stem is always green in color, old stem will be brown. It will bear leaf, branches, flowers, fruits. It will be responsible for conducting water and food throughout the plant body, right? And it bears bud also, terminal bud and axillary bud. What is the meaning of this terminal and axillary bud? You have to remember, okay, any bud which is present at the tip will be considered as terminal bud, right? And any bud which is present at the axil, leaf axil, that is called axillary bud, right? So I have drawn two buds here. This bud is called terminal or apical bud, and this bud over here is called axillary bud. What is the function of this? Terminal bud is responsible for two things. It will increase stem length, right? It is responsible for increasing the length of stem. Another thing it is doing is it is forming leaf formation. Can you see leaf primordia are present there, right? These two things are leaf precursors, and this will be making two things. Either it will make a branch or it will make flowers, right? So based on position on a stem, you can have two types of bud, terminal and axillary. Then here, this is the modification of stem which is deleted, but you can remember that there are sometimes complete stem can get modified, sometimes a part of stem can get modified, sometimes stem will be touching the ground, some part is above the ground. So there are three types of modification: underground modification, stem has become underground and become modified; subaerial modification, some part is interacting with the soil, some is in aerial condition; and aerial modification, any part of the stem which is above the ground, that if it is modified, that is called aerial. Tuber. Tuber is underground modification of stem, which is there for storing food. Actually, all are for storing food only. Tuber is found in potato, rhizome in ginger and turmeric, corm in colocasia and zameen and bulb in case of onion and garlic. Right? So this you have to remember and memorize. Then offsets may you have to remember pistia and icornia. Internode length is very short and reduced. That's why they all appear together. Right? Runners, you have to remember internode length is very long. Right? From one point, 10 new plants are created, they will go very long because next plant will be formed at node only. Right? So length between the internode is long, so the plants, new plants can reach to new niche or new habitat very easily. Right? In grasses, strawberry, oxalis, you can see this. Then we have suckers. Suckers may you have to remember banana, chrysanthemum and pineapple. Here what happens, you will see underground the stem will grow laterally for some time, then obliquely it will come out. Maybe you have seen in banana, some small banana plants will always be present near the parent big banana tree. Right? So banana, chrysanthemum and pineapple, they produce suckers. They all will act like vegetative propagation methods also. Stolon may you have to remember that uh, one branch will grow, then it will arch down, bend down. As it will touch the soil, again new plant will come. Right? So this kind of condition is called stolon. It is found in mint and jasmine. Right? So I'm not going in much detail in this because this is not there in new version of NCRT, but definitely PYQs are there. We have to remember all these examples. Total 11 type of modifications are there which you have to remember. Then aerial modifications, may you will see you have tendrils. Tendrils is what? It is a axillary bud of the stem will get modified into this coiled structure which is called tendril. In grapevine, in gourd family, you will be having these tendrils. Thorns, you have to remember citrus and bougainvillea. Right? Pointed, sharp, woody structures are there in for protecting from browsing herbivores and all. Here you have to remember thorns, and thorns will be formed. Pillowclad may you have opuntia and euphorbia. Opuntia is a flat photosynthetic stem and euphorbia is a cylindrical photosynthetic stem. The entire stem will be converted into flat or cylindrical structure. Right? Now we can talk about leaf. Leaf is a laterally attached flat structure which is developing at the node, but it is originated at the tip by shoot apical meristem. The arrangement is acropetal. Young leaf will be present at the tip and the old mature leaves are at the base of the stem. Right? The leaf is divided into three parts. This is the leaf base, this is the petiole, and this is the leaf lamina. Right? You have to remember leaf lamina, you will be having veins and veinlets. Leaf base can be modified sometimes. You will see leaf base can either be swollen or it can be sheathing. A swollen leaf base is also called pulvinous leaf base. Right? This condition is found in leguminous plants. Okay, it's a modification of leaf base. Right? And sheathing leaf base is present in banana. Okay, either it can be covering the stem partially or completely. Okay, you have to remember this. Actually, leaf base is a junction between the main stem and the petiole. Petiole is a stalk like structure, and then this entire thing is called leaf lamina or leaf blade. Right? Now we have different types of leaf: simple leaf and compound leaf. If we consider simple leaf me you have to remember, if the cutting, if the leaf is not having any kind of incision and the margin is intact, then it is called simple leaf. Or if any kind of incision is there but it is not touching the uh, midrib, it is only present up to the you can say outer part, then it is called simple leaf. Compound leaf may incision is reaching up to the midrib, dividing the entire leaf lamina into small structures called leaflets. Right? And this structure is called rachis, a common axis on which all the leaflets are attached. And here you have to remember, this is also a kind of compound leaf where at a common point all the leaflets are attached. This common point at which all the leaflets are attached is what? It is the tip of petiole. Right? These are all the leaflets which are attached here. right? So you have to remember this. This kind of condition is called pinnately compound leaf. This kind of condition is called palmately compound leaf. You can see like feather it is appearing, right? And this is like a palm of hand. Pinnately compound leaf is observed in neem, right? This is the example. And palmately compound leaf is there in silk cotton, right? Now we have phyllotaxy, arrangement of leaf on the stem. If you consider phyllotaxy, here you can see that maybe the leaves are present only one leaf at one node, right? Maybe only one leaf is present at one node. This is called alternate type of phyllotaxy, that is there in mustard and sunflower. Or maybe each node can have two leaves like this, right? This is called opposite phyllotaxy. This is alternate and this is opposite. Or maybe you can see on one node you have multiple leaves attached, right? This is called world phyllotaxy which you have in these plants, right? Alternate in mustard and sunflower, opposite guava and calotropis, and world alstonia, right? Modifications of leaf may you have to remember for supporting leaf can convert into tendril like in pea plant. For defense, leaves can convert into spine, or sometimes you can say to avoid transpiration also leaf can reduce the surface area and they can change into spines, right? For photosynthesis there is some uh, leaves which are called phyllodes. Here the petiole will get converted into a photosynthetic structure, so petiole will become photosynthetic, and this condition is called phyllodes. This happens in the Australian acacia plant because their photosynthetic leaves are very tiny and short-lived, right? Insectivorous plant may complete leaf is modified into insectivorous structure, that is a picture, right? So that is for that. So this is there in the leaf. Now venation, you have two types of leaf on the basis of what is the arrangement of veins and veinlets. You have reticulate venation where you have a network of vein and veinlet, right? And you have parallel venation where all the veins they run parallelly, right? In which plant do you have this reticulate venation? You will have this in most of the dicots, right? What about this parallel venation? You will have in most of the monocots, and in the NCRT they have given an image like this. Okay. Now if I talk about the inflorescence here, so you can see flower is a modified shoot, right? Shoot itself is modified, the axis is condensed, and instead of having different structures like leaves and all, here we have the floral appendages formed, right? So during flowering, shoot apical meristem modifies into floral meristem. There are a lot of hormonal changes also. Internodes do not along, and axis will get condensed. As a result, you will see that the shoot will be modified into flower. Okay, we can have inflorescence. This is arrangement of flower on the floral axis, right? Let's suppose this is a particular axis on which different flowers are arranged, right? So this axis will be considered as inflorescence axis, right? Or floral axis. What is the arrangement of flowers on this inflorescence axis? That is called inflorescence. So maybe a flower can be a single flower which is called isolated solitary flower. Shoot tip is converted into a flower. Let's suppose I am making a shoot tip and here I am making a flower here. So this condition is called solitary condition, right? Or maybe if the flowers are arranged here uh, under the two categories, that is racemose inflorescence and cymose inflorescence. In racemose inflorescence, we have to understand that the main axis will continue to grow, right? Because the axis will never terminate into flower, right? For example, if I draw here, it will be like this. Old flower, youngest flower, it's continuing to grow. But here what is happening? Oldest flower is convert, like the old flower is present at the tip and the young flowers are coming from the side, right? This is cymose inflorescence. It is indefinitely growing axis. Here it is having limited type of growth. Here the flowers are born on acropetal orientation. You can see old flower here, youngest flower here. Here it is basipetal. Oldest flower is present here, the young flower will be present near to the base, right? A flower may, these are the different types of parts of flower which you have studied. We have different words: calyx, corolla, androecium, and gynoecium, right? If we talk about calyx, it is made up of sepals. Corolla made up of petals. This made up of stamens, and it is made up of carpel or pistil, right? calyx का main function is it is going to protect the flower when the flower in its bud stage. This is bringing pollinators, right? This is making male reproductive structure, so it is making male gamete. This will be making female gamete, right? Now here you have to remember generally the color of calyx, it can be green, right? Okay, now it is not photo, it will not contribute much to photosynthesis. You have to remember here the calyx can show you two conditions: all the sepals are fused together, that condition is called gamosepalous, right? All or the sepals remain free, that condition is called polysepalous. Here all the petals can remain fused, that is called gamopetalous. Here all the petals can remain free, that is called polypetalous, right? So these two terms you have to remember. These are the morphological terms which you have to memorize, right? Androecium may you have to remember, if you take one stamen is divided into two parts, anther and filament. The tip part is called anther, and at the towards the base we have filament. Each anther is having two lobes, right? This is the anther, right? You will have anther like this, right? If we cut it, we'll see a structure like this, right? So this is one lobe, this is second lobe. These are the two lobes of anther. Each lobe has two chambers, right? Here you have these two chambers in each and every lobe, right? A, a particular stamen which is sterile, which will not make any particular pollen, that is called staminode, right? Then here gynoecium, you have to remember it is made up of one or more carpel. Number cannot be fixed, either only one carpel is there or multiple carpels are there, right? Carpel is actually made up of three parts, if you remember, right? It is having these three parts. You have a ovary, you have a stigma, right? And you have style. So you have stigma, style, and ovary, right? Three parts are there. Each ovary bears one or more ovules. Inside this ovary there are ovules, right? Here there are structures which are called ovules, which are present here within this, right? Or we can draw it like this. Okay, ovules are present in this structure, connected to the wall of ovary with a flat cushion like structure called placenta. Carpels, if they are multiple in number, all the carpels can remain free. For that we have a word called apocarpus. If all the carpels are fused, then we have another word for that, that is syncarpus. They have given these examples. Lotus and rose is an example of all the members where you have carpel as free, lotus and rose. And for united or syncarpus, you have tomato and mustard. So you have to remember this. Sometimes we have to remember within a flower when we cannot differentiate that the structure which we are looking at is calyx or corolla, right? At that time we will be using a term which is called perianth for it. Perianth is having an individual structure which is called tepal. So when we are not able to differentiate between this calyx and corolla, we will be stating it has another word that is called perianth. It is made up of repeating unit which is tepal, and in lily you will have this, right? Now aestivation, what is the arrangement of a petal or sepal with respect to another petal or sepal in a floral bud, right? That is called aestivation. Now see here, there are different type of aestivation: valvate, twisted, imbricate, and vexillary. If you see here, first thing they will be asking you will be these different you can say examples which they have given. You will have valvate condition in calotropis, you have twisted condition in china rose, ladyfinger, cotton, cassia, gulmohar, you will be having imbricate, and pea and bean you will be having vexillary condition. What is this? Actually if you see in bud condition, when you will try to check the base level pe what is the arrangement of petal or sepal, you will be clearly able to understand this. In valvate type of estivation, can you see if this is one petal, it is touching the other petal, then it is touching other petal, other petal. Here the petals are touching each other just at margin with zero overlapping. There is no overlapping at all, right? You can see very well that there is no overlapping in between the margin of any petal, right? So when there is no overlapping, just end to end the petals or sepals are touching each other, that is called valvate estivation. But in this figure what you can see here, you can see that uh, it is twisted estivation. Twisted estivation you can see there is overlapping in between different types of petals, right? And this overlapping is in a regular fashion, right? You can see that here this is internal, this is external, this is internal again, but next one ka it is external again, this is internal, this is external. So when there is overlapping between the ends of the petals or margins of the petal, and that overlapping is in the regular fashion, then it is called twisted. You will get to see this in China rose, lady finger, and cotton, right? Imbricate estivation, maybe overlapping is there, but this overlapping is not in a regular fashion. You can see one is completely internal, see their margins, completely internal, right? This is completely external, see their margin, and these three are twisted, right? So there is not a regular fashion overlapping, right? Here a regular fashion is followed, a ordered overlapping is there. Here it is unordered. Two are, one is completely internal, one is completely external, rest three are twisted. This you will have in Cassia and Gulmohar. Vexillary estivation, when you will see, this is a specific type of estivation which you are going to see in only P and bean. Here one posterior petal is present, that is called standard, right? This standard is largest. This standard is going to overlap these two which are called laterals, right? Laterals or wings, right? And these wings or laterals are further overlapping the two fused petal, that is called keel, right? So this, when you will write in floral formula, you will write C1 plus 2 plus 2 which are fused. This is standard, this is lateral, these are two fused keel, right? So this is the estimation which you have to remember. They will always ask questions on example. Along with that, you should know what is happening in this. Now if I talk about a specific flower, I have detailed everything summarized here. There are so many technical terms which are there used in the flower, so you have to remember that. If I talk about a flower, flower can be of two types. Either it can be unisexual or it can be bisexual. Unisexual means either a flower is having stamen or a flower is having pistil, right? When it is having a stamen, right, when it is having androecium, I will call that flower as staminate flower. It is not staminode. Staminode means it is sterile flower. It is androecium is there, it is staminate. Only carpel is there. If it is only having carpel, right, then you have to remember that kind of flower is called pistillate flower, right? Female flower. Only either this is there or this is there, right? Either male reproductive structure or female reproductive. Bisexual flower is having both stamen as well as carpel, right? So two types of flower based on their sexuality, you understood. Now if I take one flower, okay, and I start counting how many petals, how many sepals, how many androecium, how many gynoecium is present, then I will be talking about isomery of the flower. So a flower can be trimerous, right? Here all the floral appendages are in multiple of 3. For example, I may have 6 petals, I may have 6 sepals, I may have 6 stamens, right, or 3 carpels, right? So it is multiple of 6 or not, that is trimerous. In most of the monocots you will have this condition. Monocots may, this is common, I'm not saying always present, but it's commonly occurring there. Tetramerous and trimerous, this is common in dicots, right? It is commonly occurring in dicots, right? What you have to remember here, what you have to remember here, that tetramerous may everything is there in multiple of 4, pentamerous everything is in multiple of 5. 5 petal, 5 sepal, okay, 10 anther, right? Then 5 carpels, like that you have to remember this. Mostly gynoecium does not follow this character, it will be following mostly up to androecium part only. Now this we understood, this we understood. Now let's talk about symmetry. Symmetry means, can you cut a flower and divide it into 2 mirror images, right? If yes, then the flower will be considered as symmetrical flower. If no, then the flower will be considered as asymmetrical flower. Uska example is Kana lily. Okay, some flowers can be divided into 2 equal half from a single line passing through its center only. That flower is called zygomorphic flower. It can have bilateral symmetry. If you can divide the flower into 2 equal half from any line passing through the center, then it is called radial symmetry or actinomorphic symmetry. Just say for example, if I draw a flower like this, right? This flower I can divide into two equal halves only by one single plane passing through the center. This kind of flower is there in P, Cassia and Gulmohar, right? But if I have a flower like this, right, I can divide it in equal half like this also, this also, this also, this also. This kind of flower is called radially symmetrical flower. This is there in chili, mustard and datura. right? So this is also completed. Now types of flower, if I consider, right, if I consider the types of flower on the basis of the position of ovary, what is the position of ovary with respect to other floral whorls, I may have three conditions: hypogynous, perigynous, and epigynous. If you see this image which I have made here, which is I have kept here from NCERT, in this image you can see we have three conditions given here. This one is hypogynous, these two together perigynous, right, and this one is epigynous, right? What is happening here? See the position of ovary with respect to other floral appendages: sepal, petal, and rachim. Here also sepal, petal, and rachim. Here also same thing, right? With respect to sepal, petal, and stamens, can you see ovary is superior, right? So in this case, what you have to remember here, ovary is superior, right? Ovary is superior, other floral whorls are coming below the ovary, right? This kind of condition is called hypogynous condition. This is found where? This is found in mustard, china rose, and brinjal, right? Ovary is superior, other floral whorls are coming below the level of ovary, right? In this perigynous, can you see what is happening here? Or first I will tell you this. Epigynous, just see here, other floral whorls are present above the ovary, ovary is below everything, right? So you have to remember here what is the condition. Here ovary is inferior, understood? Ovary is inferior, and just notice the point of attachment of sepal, petal, and stamen. They are attached above the level of ovary. This condition is called inferior ovary. This you will get to see in ray florets of sunflower, in guava, and in cucumber, right? So three examples are there. Every time they have asked PYQ on this, right? Guava, sunflower, and cucumber, you have this condition. Okay, if this is superior and this is inferior condition, what is this? These two are telling us about the perigynous condition. Here you just try to see what is this structure called. This structure is thalamus, right? Thalamus or receptacle on which all the other floral structures are present. So if you see the thalamus or receptacle, can you see the thalamus has tried to grow upward and try to make the ovary inferior, right? But it failed, right? It failed, and all the other floral whorls are present at the rim of thalamus. All the other floral whorls are present at the rim of thalamus. If you see here, okay, then this condition is called what? It is called ovary is half inferior. Okay, half inferior, and you will see this in plum, peach, and rose. You can see here plum, peach, and rose. Here gynoecium situated in the center, other floral buds are located at the rim of thalamus. So ovary tried to become inferior but failed. So everything was at the margin of the thalamus only, right? So this you have to remember, the types of you can say flower based on the position of ovary. So we have understood different types. Okay, isomerous flower, unisexual, bisexual, hypogynous, everything we understood, and symmetry also we have understood, right? So this is a huge part which we have completed. Now there are certain things which are given with the respect to stamen also. If we consider the male reproductive part stamen, right, we can see two conditions. Stamen can be attached with floral whorl, right? It can attach as with petal in the condition called epipetalous, whose example is brinjal, or it can attach with the perianth, whose example is like whose example is lily, which is called epiphyllus. Just say for example, if I'm saying that this is the tepal or petal, and I'm saying that this is anther filament part. This filament part is attached like this, right? First, the filament part will be attached on the thalamus. Then the lower part of the filament is attached or sticked to the tepal or petal also, right? So when the filament is attached to the petal, it is called epipetalous, and we see this condition brinjal. If it is attached to the you can say in the perianth, right, that is called epiphyllus, and it is found in lily, right? So you have to remember like this, that if this is a filament, right, and this filament part is attached like this here, right, then this condition is called epiphyllous. If this is tepal, and we have this in lily, right? And if the same thing I draw here, and if I write that this thing is not tepal, it is a petal, right? So this is what it is, epipetalous condition, and this we are seeing in brinjal. Understood, right? Then we have attachment of this stamen with each other. Sometimes these filaments can attach among themselves. Sometimes they are attaching to other structures like a petal or a sepal, sorry, a petal or a tepal. Sometimes they are attaching among themselves, right? If they are attaching among themselves, then there are conditions like monoadelphous, diadelphous, right? And polyadelphous, right? Three conditions are there. Okay, this is the first one. Maybe I have to number them: monoadelphous, diadelphous, and polyadelphous. Monoadelphous means, let's suppose all the filaments are joined together. Remember, you have seen China rose. All the filaments are joined together lengthwise, right? And they are only free at the tip like this, right? And you will see anthers will be present here, right? This kind of condition is called making one single bundle, all the filaments are fused, right? One single bundle mein all the filaments are fused. This is called monoadelphous. You will find this condition in China rose. When the same happens and you have two bundles like this, okay, when you have same thing but two bundles like this is called diadelphous, and this happens in pea plant mein. This happens, there is 9 plus 1 condition, right? 9 and ratium are joined together in one bundle, another is single, right? More than one, more than two bundles if it is present, then it is called polyadelphous, and this is there in citrus. Okay, in citrus plant you have this condition. Polyandrous means all the filaments are free. When you have a flower and all the filaments are free like this, they are not attached, they are free, then this condition is called polyandrous. Example is not given in NCERT. Okay, so you have to remember. Okay, then within the ovule, what is the arrangement of sorry, within the ovary, what is the arrangement of ovules? That is called placentation. With the help of placenta only these ovules are arranged. So based on that, we have marginal, axial, parietal, free central, and basal. Very very important, you have to remember their example. Pea is an example of marginal placentation. China rose, tomato, and lemon, you have this condition. Mustard, argemone, parietal placentation. Primrose, dianthus, free central. Sunflower and marigold, you have basal placentation. Can you see here what is the condition here? These are different ovules. Can you see these ovules are present on the ridge formed by, okay, these are arranged on the ridge formed by what? Ventral suture. Two openings are there. One opening from the pea pod by which you can open it, that is called dorsal suture. Opposite to that, there is a ventral suture. There is a ridge which is formed by ventral suture. On them, if the placenta is born, that is called marginal placentation, right? Axial placentation, what you have to remember, one more thing, this is a unilocular condition. Here you can see when multiple carpels are fused together, all the carpels are actually joining in the center. This position is called axial position, right? This position is called axial position. When all the septa are joining in the axial position and the ovules are born on the placenta which is attached on the axial position, it is called axial presentation. Parietal presentation, you have to remember, when all the ovules are connected on the peripheral part, right, at the peripheral part, then it is called parietal presentation. On the inner wall, on the peripheral part, all the ovules are attached. Mustard and argemone, you have this condition. Initially this is a unilocular ovary, but there is a formation of a false septa, so it will become bilocular, right? So you have to remember this. Here pre-central presentation, you have to remember here what is the condition. You can understand it very easily when you take a notice of this. There is a parenchymatous mass of tissue which is present like this, and if I'm saying that all the placenta is born on this, right, and the ovules are attached here, right, like this. If I cut the structure and I see, it will appear like this, right? So ovules are born on central axis, right? Ovules are born on the central axis. Okay, this is that central axis on which all the ovules are attached, which is made by the parenchymatous tissue, right? Primrose and dianthus may you have this condition, right? Here also you have unilocular. This is unilocular. Initially unilocular will become bilocular because of false septa formation. Here always multilocular. This is also unilocular. Here you can see that placenta is actually born at the base of the ovule, right? And a single large ovule is present here. This is called basal presentation. In monocots you have this, in grasses you have this, sunflower, marigold you have this condition, right? Now the last part of the chapter, see family part, you will study by yourself, you revise it, right? If any time permits, any extra session is there, we can do the families, but just by technical terms itself it will be very easy for you to remember all those things. So we'll be revising only the theory part. Family part we have not added up here, right? So here you can see next is fruit and seed. After the fertilization, the flowers will be transformed into post reproductive structures, which are fruit and seed. So see, ovule after the fertilization is going to become a seed, and the ovary after the fertilization will be ripened into the fruit. Seed contains two things. It will be having embryo and it is having the endosperm. Embryo is a future plant and endosperm is a continuous nutrition for this embryo, right? Endosperm is a continuous nutrition for this embryo. If you will see the seed, seed will be having seed coat on top, which is divided as testa and tegmen. The outer seed coat is called testa, inner seed coat is called tegmen, or you can say the inner seed coat is going to convert into tegmen, outer seed coat is convert into testa. Fruit is divided into seed and pericarp, right? When you will see a fruit, you will be seeing a fruit wall and you will be seeing seed, right? So this pericarp is further divided into epicarp, mesocarp and endocarp, right? Which is the outermost here, mesocarp middle, and inner is this. Up this pericarp or fruit may be fleshy or it may be dry fruit, right? If it is a fleshy fruit, it will be mango, it will be guava, it will be guava, it will be orange, it will be fleshy fruit. Dry fruit में groundnut, mustard, all these are dry fruit, right? Ab they will always ask a question, mango में what is the edible part? Edible part or edible layer of pericarp is mesocarp, right? Okay. Coconut, what is edible? In coconut you have to remember none of the layer is edible. Instead, endosperm, the nutritive tissue, is edible here, right? This you have to remember for these two things. And then you have to remember these two are actually example of a fleshy type of fruit, which is called drupe fruit, right? Drupe fruit is always from the formed from a monocarpillary ovary. Okay, so you have to memorize all these things. Okay, now this is the dicot seed and monocot seed structure and detail. Okay, dicot seed name itself is because it's having the two cotyledons. Cotyledon is that structure by which the endosperm nutrition is absorbed to the embryo, right? Here a shield shaped single cotyledon is present in monocot seed, that is called scutellum, which is actually not shown in the diagram, but you have to remember here laterally attached one structure is there, which is called the labeled as scutellum here, right? Here in dicot seed at maturity, there is no endosperm except castor, right? Except castor, there is uh, no endosperm. Castor is dicot, still it has endosperm. Okay, monocot seed is endospermous. Okay, orchid is a monocot, still it does not have any endosperm. Embryo consists of embryonal axis, radical plumule, right, and two cotyledon. Embryonal axis is there. The tip part of this is called plumule, upper tip, lower tip will be radical, and two cotyledon is there. Here you have to remember there is a seed coat here, right? Outer seed coat is testa, inner seed coat is tegmen. Outer is formed by outer integument, inner is formed by inner integument. There is a small scar on the seed coat that is called hilum, which is situated above the you can say which is present. Now above the hilum there is a small opening or a pore that is called micropyle, through which oxygen can enter, through which water can enter, right? So here you can see this is a micropyle and this is hilum, right? In this you have two cotyledons, you have radical, plumule, everything is there, right? Here you have to remember you don't have a seed coat here. In the case of monocot seed coat is not that much thick and not divided into two layers. It is membranous, it is fused with the fruit wall, right? And endosperm is bulky here, lot of endosperm is there. Outermost layer of endosperm is called aileron layer, it is proteinaceous layer, right? It separates this endosperm from the other component of the you can say seed, right? And the the particular protein which is present here is amylase protein, right? Amylase protein is present here, right? So it will convert the starch into glucose and it can easily get mobilized to the scutellum, right? So this was all. The family part I have not added here because the session was actually exceeding the time. So if any time will be there, definitely families we have to do, right? Now roots originating from parts other than radical, they are called, okay, radicals is the root which is getting formed is called tap root. Any root which is formed other than radical is called adventitious root. In gods, axillary buds develop spirally coiled structure called, right? So that is called tendril only. This is spirally coiled structure which is there in the bottle gourd and all Cucurbitaceae family. Pulvinous leaf base found in leguminous plant, right? The solen leaf base is found in leguminous plant. In Australian acacia, which one is present? Phylloid, cladode, clad. So it is phylloid, photosynthetic petiole, right? Now choose the mismatched pair of leaf character with its example. What is mismatch here? Opposite phyllotaxy in calotropis? Yes. Palmately compound leaf is Alstonia? No. What was the example of palmately compound leaf? Silk cotton, right? Alstonia and Nerium has world type of phyllotaxy. Alternate phyllotaxy channel was correct. Leaf tendril is found in T, correct. Right? Now, which of the following is incorrect match? Perigynous flower. Perigynous means half inferior. Plum, peach, and rose, correct. Monoadelphous condition, P, wrong. P is having diadelphous condition. Remember, monoadelphous is there in China rose, right? Epigynous flower, guava, cucumber, ray florets, correct. Polyadelphous, citrus, yes. Okay. Racemose inflorescence is identified by what? In racemose inflorescence, what happens? Main axis never convert into flower. This is the young flower present at the tip, old flower present at the base. So acropetal, young at tip, old at base, yes. Presence of sessile flower, no. Continuous growth of main axis, yes. So can we choose A and C? Yes, we can do it. Correct position of floral parts over thalamus in mustard plant is in होगा यहाँ पे, ठीक है? It is in, right? So you know very well mustard plant will follow which condition? It is having hypogynous condition. What is the meaning of hypogynous condition? Hypogynous means superior ovary. Just find. Gynecium situated at the center, other part located at the rim. Rim means perigynous. Gynecium occupies highest position, other plant situated below it. Yes, this is the answer. Rest, we should not waste our time. You can read by yourself, right? Placentation in which ovules develop on the inner wall of ovary or peripheral part, right? Inner wall of ovary or on the peripheral part, it is parietal placentation. Can you tell me the example? What are the example of parietal placentation? Argemone and mustard, right? Which of the following plant shows maxillary estivation and diadelphous stamen? That condition is there in Pisum sativum or pea plant, right? Fabaceae, you have to read, right? Now the last chapter, that is anatomy of flowering plant, right? We have done 3 chapter revision. Now the last chapter, that is anatomy of flowering plants, we have to revise. You just tell me what's the meaning of anatomy. The study of internal structure of anything is called anatomy, right? So when you will take up the plants, you have to study the tissue. What is the meaning of tissue? Group of cells, right, which performs similar function, right, and they have origin which is common. So common function they should perform, common cells they should have originated. In the plant cells, you have to remember we have different type of tissues, right? In plant cells, how many type of tissues are there? Sorry, in plants, how many types of tissues are there? Plant body is made up of different type of tissues. The very first type is called meristematic tissue, right? Another type of tissues are called permanent tissues, right? Name itself is suggesting you. Here, meristematic tissue में division capacity is present. The cells will be dividing. Permanent tissue, division capacity lost. The cells will be there performing a specific function. All these kind of tissues will actually develop by following three things: differentiation, de-differentiation, okay, and re-differentiation, right? If you take a plant, okay, if you take a plant, उसमें the main things which are there as an organ in which you can study the tissue is three only. You have leaf, right? You have stem, and you have root. देखो, this chapter was very extensive chapter, but now they have deleted the different types of tissues, meristematic tissues and permanent tissue, because it was already there in junior classes, right? So usually when we will take this, we will teach this, but because we have less time, we have to focus only about the transfer section of leaf, stem and root. When we cut this, what kind of tissue we are going to see? Definitely we are not going to see meristematic tissue, we will be seeing permanent tissues only, right? Okay, so this we have to study as a whole. All of this will be formed from permanent tissue. This will be formed from the activity of this dividing tissue, right? And all the permanent tissues are formed from this redifferentiation and differentiation process, right? So I want to tell you about that only. Okay, देखो, very fastly I can tell you that, let's suppose I'm talking about one embryo, right? Can you tell me embryo will have any kind of meristematic tissue or not? Yes. In embryo, you will be having shoot apical meristem as well as you will be having root apical meristem, right? Can I say the shoot apical meristem and root apical meristem, they are present since the birth of the plant? So origin wise, can I call them that they are primary meristems? Yes. They are primary meristem. They are dividing in nature, right? The cells here will divide. If I draw them like this, right? So this embryo will always remain embryo only if the cells in these two regions are not dividing. The cells are dividing here, the cells are dividing and getting added up here, then only we are able to see different organs in the plant, right? So cells are dividing here, shoot apical meristem and root apical meristem. They are apical meristems is present since birth, so they are primary in origin, right? Now what we have to do? Now I'm going to write here, whenever a primary meristem, okay, अब you tell me, primary meristem में division capacity is present? Yes. Can cells divide? Yes, all the cells are actively dividing. They have thin cell wall, they have a dense cytoplasm, they have a big nucleus. Their work is to divide only, right? So here the cells are dividing, right? After division, they should make something, right? What they will make? Daughter cells. The daughter cells will undergo a process which is called differentiation. The daughter cells which are formed after primary meristem ka division, they will undergo differentiation, right? In this differentiation process, what will happen? In this differentiation process, all those cells which have divided, right, they will become specified for a specific function, right? They'll be doing certain specific function. They will become highly specific for performing that function. They will do that? By changing themselves. They will change their structure, their physiology, their biochemistry. Remember, I told you in plant growth and development chapter, right? And what primary meristem is going to form here, that will be called as primary permanent tissue, right? Primary permanent tissue. This primary permanent tissue may division capacity is lost. They can't divide, right? They will be maybe epidermal cells, maybe mesophyll cells, right? Maybe some other part of the plant body, right? So they will be there for performing a specific function. So primary meristem cells divided, they became differentiated and they became primary permanent. It can be primary xylem, primary phloem, it can be cortex, any cell, right? Now, this primary permanent cell, primary permanent tissues, इसका function क्या है? Their function is primary growth. Whatever you see in the plant body, new leaf is there, its structure is made up of what, root is there, that all is primary growth, right? Primary organ formation, that will be done by primary permanent tissue. Okay, it will be also responsible for increases length of plant. Understood? There are so many other things also, but we'll stick to here itself. Plant length will never increase if primary permanent tissues are not getting formed. Ab length has been increased, right? Now the problem will be there, that if a plant is just increasing the length, what will happen? Plant will droop down or not? Plant needs mechanical support strength also. Plant needs to increase its width, right? So what will happen? Some cells of this permanent tissue, right, they will enter in a process which is called de-differentiation. Matlab, whatever differentiation you have done, remove it. Division capacity was gone, bring it back, right? Some of the cells which are primary permanent tissue ka cells, some of them only, not all, some of the layers of those cells, they will form a meristem which is not present since birth, which is present, which is formed now, right? Here the division capacity is regained, right? Some cells will become dividing again, right? And they will divide and they will form secondary permanent tissues, right? And they're forming this by re-differentiation. Matlab, abhi you remove the differentiation process, now again do the differentiation. So division capacity again lost. They will become specific for performing a specific function only, right? Division capacity lost. Okay, what is the purpose of this? What is the function of this secondary permanent tissue? Function is secondary growth. What is the meaning of secondary growth? Increase width or girth of the plant. Understood? So three processes act together in the plant: differentiation, de-differentiation, and re-differentiation. Because of these three things simultaneously, we can see a plant growing. First, it will increase in length, then it will start increasing in girth, right? For increasing the length, the meristem is present in the beginning. For increasing the girth, we have to develop the meristem by a de-differentiation of some of the primary permanent cells, right? They will regain the division capacity, they will become meristematic and they will start dividing, right? Understood? How you will understand this? See, if this structure has to increase the length, easily it can do or not. New cells will be added in this direction, the length will increase, right? Because we have dividing cells here, there is no problem. But if you want to increase its girth like this, what will happen? The cells which are present here in the center, all are primary permanent cells, right? All are primary permanent. Nothing divides here. Meristematic cells are only here at the tip. Nothing divides in this area. And if I want to increase the girth, I need some layers to divide. Then only this entire structure will increase in size, right? So what will happen? Some of the cells which are present here, they will become de-differentiated and they will become secondary meristem. If they will become secondary meristem, they will be adding new layers of the cells on both the directions, right? And if they are adding new layers of cell in both the direction, this layer increasing in girth, so it will increase in width or not, right? So this is what we have to study. Now, right now, one more topic is there, that is called tissue system, in which we have epidermal tissue system, ground tissue system, and vascular tissue system, right? So I'll be discussing about the tissue system here very quickly so that I can go on to the section and we can complete this. Okay, tissue system. Three basic tissue systems are there: epidermal tissue system, ground tissue system, and vascular tissue system. ठीक है, epidermal tissue, entire plant body can be formed by these three things. If I consider epidermal tissue system, I am having here epidermis, which is usually single layered structure, right? Sometimes it is multi-layered in nerium and all, but usually it is single layered. It's protective in nature. Cells don't have any chlorophyll, right? And then we have stomata, right? Stomata may we have three things, right? In the stomata, we have subsidiary cells, guard cells, and stomatal aperture. Together, all of this makes stomatal apparatus. You have to remember, only guard cell is a specialized type of epidermal cell where you have chlorophyll. Otherwise, no epidermal cell has chlorophyll. There is no intercellular spaces between the epidermis also. Parenchymatous cells they are, right? Very less amount of protoplasm is there with a very big vacuole in the center, right? Stomata is present as a pores in this epidermis which is there in the leaf or maybe in the young stem, right? Stomata is having three different you can say structures, I already told, right? You have to remember the shape of stomata. In case of sorry, shape of guard cells of the stomata. In case of your dicot plants, you have a normal kidney bean shape stomata guard cells, and in case of your monocots, we have dumbbell shaped guard cells, right? You have to remember all these things. In case of guard cell, the outer walls are always thin, inner walls are always thick, right? Then we have some stomatal aperture appendages like trichomes, and we have root hairs, right? So if it is root epidermis, you have to remember the extension is called root hair. It is a unicellular structure. It's made up of only one cell. Its extension of plasma membrane will make that hair. And trichome is what? Multiple cells are joined together to form this multiple cell ka structure, that is called trichome. Trichome is stiff, soft. Trichome is having different type of functions. It is secretory sometimes. It is preventing the water loss, right? Root hairs on the other hand, it is increasing the absorptive surface area for the plant. So these are the things which you have to study for epidermal tissue system, right? Ground tissue system mein you have to remember everything which is excluding epidermis and the vascular tissue system, what is left that will form ground tissue system, right? So it is like it starts from cortex, right? Cortex is there, then last layer of cortex, that is endodermis, is there, right? Pith is there, pericycle is also there, right? Pericycle is there and pith is there. अब you have to remember this is only made up of parenchyma, right? Epidermis is always parenchymatous, living thin walled cells. This is made up of all types of simple tissue. Remember class 9th you have studied that simple tissues are made up of three types. Okay, parenchyma, collenchyma and sclerenchyma. All three are possible here in different sections, right? And vascular tissue system, you have to remember, it is made up of complex permanent tissues. Up here they have mentioned about the type of vascular bundle. If you will see vascular bundles, you will be seeing different types, right? Sometimes you will see vascular bundle where you have a phloem over here and a xylem over here. Sometimes you will see you have a phloem over here, you have one more layer and you have xylem here. And sometimes you will see that you will be having vascular bundles which are separate, right? This type of vascular bundle where this layer is there, which is called cambium, right? This is called conjoint closed vascular bundle. This is called conjoint open vascular bundle. Okay, cambium means it is going to do secondary growth, right? So this conjoint open is there, conjoint close is there. Then we have one more where you have radial arrangement of vascular bundle. Let's suppose you have xylem over here, right? So you'll be having phloem over here, right? This kind of arrangement is called radial. Okay, this vascular bundle you will get conjoint close, you will get in all the leaves. You will see this in monocot stem. This you will get to see in all the dicot stem. And this one you are going to see in roots, all type of roots, whether it is monocot or dicot. Here you can see the vascular bundles are arranged in two different radii, separate radii, independent bundles are there, right? Here they are jointly present together, right? Cambium is present, then it is dicot stem open to secondary growth. Here cambium absent, closed vascular bundle. Okay, conjoint closed vascular bundle. Abhi cortex also can be differentiated or it can be undifferentiated. In the root you will see undifferentiated cortex, in the stem you will get differentiated cortex, in the dicot stem you will get well differentiated cortex, and in the monocot stem you don't have any particular differentiated cortex, right? So I will discuss about all of these things in the sections. Let's begin the sections. Okay, so this is the very first one where you can see we have to study the anatomy of मोनोकॉट रूट एंड डाइकॉट रूट. देखो ये डाइकॉट रूट है, ये मोनोकॉट रूट है, right? Just try to pause the video and see by yourself, can you see any particular difference in both the roots, right? For dicot root they have used sunflower root. Please remember this is a primary root, secondary growth has not been done, and for this they have written that they are taking sunflower ka root, and you must remember sunflower belongs to family Compositae, right? So it's a dicot, right? Here you can see root hairs are there. In the next section also you can see root hairs are there. Number is little bit less or more, but that does not matter, right? It's a extension of epidermis, root hair is there. Okay, root epidermis is also called epiblemma. It is not mentioned here, but you have to remember it is called epiblemma, right? Root epidermis is also called epiblemma. So this single layered epidermis you can see here, single layered epidermis you can see here also. Then can you see the green color cells which are present here, right? The round cells with some few intercellular spaces. The cells are parenchymatous here, thin cell wall containing cells, right? Same condition here, right? This is called undifferentiated cortex, which is there same in monocot as well as dicot root. Then you have to remember they have shown us endodermis, right? Ab dekho, cortex, if it is differentiated it must be having three layers, right? Hypodermis, hypodermis, general cortex, also called cortical layers, right? And the last layer of cortex is endodermis, right? So in the root, what do we have? Do we have hypodermis? No. Do we have general cortex? No. We only have endodermis, last layer of cortex, right? Yeah, we have this general cortex also filled here up to here, right? So this kind of cortex will be called undifferentiated. Cortex is not differentiated into its three layers, right? It's having last layer endodermis, and it is having just a, just you can say general cortex, which is packed with the parenchyma cells, right? So that is why this kind of cortex is undifferentiated. So last layer of this cortex, it is called endodermis. If you see here, they have shown it here, right? This endodermis is a single layered, you can say layer, which is having which kind of cells? Barrel shaped cells. Okay, they have barrel shaped cells. Okay, the cells are having thin cellulose containing cell wall. They have suberin deposition in them, right? Suberin is deposited in both tangential and radial wall, right? As a result, it acts like a checkpoint. Nothing can enter in this, right? No pathogen can cross it. Its suberin is actually non-polar type of material. It is hydrophobic, right? So it will be present here. Barrel shaped cells are there. Then next to endodermis, there is pericycle. Pericycle is very important layer, because pericycle in dicot roots, pericycle cells will divide and they will form lateral roots. In monocots also, pericycle cells will divide and they will form lateral roots. But in dicot, additionally, pericycle layer will get de-differentiated to form the vascular cambium, right? So pericycle is involved in vascular cambium formation plus lateral root formation, but on the other hand, here the pericycle is only responsible for making lateral roots, right? Endodermis is barrel shaped here also. Endodermis is having suberin here as well, right? Then there is a tissue which you can see here, which is in the center, right? That is called pith. That same pith is present here. Parenchymatous cells are making that pith, right? In this section, everything is parenchymatous except some suberin deposition in the cell wall of endodermal cells, right? And the shape is also very, pericycle is having thick wall parenchymatous cell, right? Understood. Then you can see here they are showing vascular bundle. You have to remember these small cells which they want to show you here. This is phloem, right? And these cells which they want to show, this is xylem. So can you see xylem? Xylem is present as a separate bundle, phloem is present as separate bundle. This condition is called radial type of vascular bundle. Vascular bundles are radial here, right? Can you see here protoxylem is actually present here, metaxylem is present here. When the protoxylem is facing the periphery, metaxylem is facing the center, this kind of condition is called exarch. In all the root, you have exarch type of condition of the primary xylem, right? Up, this is a dicot root. So you have to remember here what is the condition. Here we have pith, which is poorly developed, right? Pith is poorly developed, right? Pith is very poorly developed. And if you count the number of vascular bundles, okay, how many are there? Only 2 to 4, right? So it is called diarch to hexarch type of condition, right? And you can have a tissue here in between the xylem and phloem. That tissue which is present in between xylem and phloem is called conjunctive tissue. This is made up of parenchymatous cells, okay? Conjunctive tissue. Conjunctive tissue and pericycle cells both will help in vascular cambium formation, right? So this is the condition here. Same is present here also. Only difference which you can get to see is here the number of vascular bundles are not 2 to 4. It is more. Vascular bundles are more in number, and this is called polyarch condition, right? After that, you have to remember here the pith is more developed, although it is not visible here in the diagram of NCRT, but you have to remember pith is much more developed here, right? Vascular bundle is polyarch. This condition is different. Here the pericycle, it only forms lateral root, but here the pericycle, two things: lateral root Remember secondary root, tertiary root, all that. And vascular cambium. Understood? Only that is a difference. Now we can see anatomy of dicot stem and monocot stem. So this is the overall arrangement of dicot stem. What are the important things you can get from here? Can you see vascular bundles? They are arranged in ring-like arrangement, right? Here, can you see they are arranged like ring here, right? One vascular bundle is zoomed in here and shown. Can you see the vascular bundles here? The vascular bundles in monocot stem, they are actually scattered. They are not arranged in a ring-like fashion. They are scattered, right? Then if you see here again, can you see that here the after epidermis is shown? After epidermis, can you see hypodermis is shown? And then after hypodermis, they have shown us this parenchymatous cortical layer, right? And they have shown endodermis. Can we say here cortex is differentiated? Yes. Cortex is well differentiated because the three layers of cortex, sclerenchymatous hypodermis, sorry, uh, collenchymatous hypodermis, parenchymatous cortical layer, and you can say parenchymatous endodermis is present in the dicot stem. So it is having well differentiated cortex. But as you come see here also you have seen that here you have collenchyma, parenchyma, and this endodermis. This is what? This is the total three layers of cortex. Outer we have epidermis. Sometimes it can be surrounded by cuticle if it is a young dicot stem, or some trichomes also you can see here, right? Those multicellular structures which are root stem hairs, right? If you just compare this with this structure, you can see hypodermis is present, ground tissue is present after this, okay, which is parenchymatous in nature, right? There is no endodermis, right? So this kind of cortex is undifferentiated cortex, right? This is undifferentiated cortex. It is not a completely differentiated cortex. Another thing, let's one by one we'll talk. After this we have pericycle, right? So you have to remember pericycle is present in semi-lunar patches, right? Pericycle is present in semi-lunar patches. And endodermis is also very important because it is having starch grain deposit in it. That's why it is also called starch sheath here in dicot, right? Endodermis is having lot of starch grain in it, so it's called starch sheath. Whereas pericycle is actually semilunar patches, which is sclerenchymatous in nature, which is just present above the phloem, right? And then you can have conjoint open type of vascular bundle. The tissue which is present in between the vascular bundle is medullary ray, and you have a very large pith here, right? You can see same things here also. You have this pericycle, you can see endodermis cells are here which are having this starch grain in that. That's why it's called starch sheath, right? Semilunar patch, you have phloem, cambium, and you have this particular you can say xylem. Now just check the arrangement of xylem. This kind of xylem in all the stems, you have to remember, when the protoxylem is towards the pith and metaxylem is towards the outer side, it is called endark condition. Every stem, whether it is monocot and dicot, they have endark condition, right? And you have this pith here. Now just try to see these same structures here, right? Can you see here pith is absent? Pith is absent here. There is no pith. Can you see there is no pericycle, right? Pericycle is absent. There is no endodermis. Endodermis, so I already wrote there, right? Endodermis is also absent. So there is no endodermis, no pericycle, no pith. That's why this is called astellar. There is no stele, right? Atectostele also it is called. Stele is not present. That by vascular bundles are spread out everywhere, right? Understood? And can you see the size of vascular bundle in the center is large, and when you are moving towards the periphery it is decreasing in size, right? And they have certain specific conditions. You have to remember that they have water containing cavities. Water containing cavities are present. right? Plus, they don't have they don't have secondary phloem, that is absent here. Okay, secondary phloem is absent here. Fibers are not there. Okay, now leaf. If you check dicot and monocot leaf, both are very different from each other. Attachment of dicot leaf is like this. Okay, attachment of monocot leaf is mostly grass and all is like this, right? If a leaf is attached like this, one side will receive more sunlight, another side will receive less sunlight. This side will be considered as adaxial and the other side is considered as abaxial. Two different sides. That's why it is called dorsiventral type of leaf. Adaxial, you can call it as dorsal side, right? This one you can call as ventral side. This all happens because of the attachment. Attachment is like this. It will receive more sunlight. This one will receive less sunlight, right? Here you have to remember this may get equal amount of sunlight from all the sides, right? Because it is getting equal amount of sunlight from all the sides, it's having only one type of side. That's why it is called isobilateral type of leaf. There is no distinction between the two side, right? It's isobilateral type of leaf here. Now you have to remember, if it is receiving more sunlight one side, it will develop more chlorophyll, right? So that's why there is a differentiation in the mesophyll tissue. The tissue which is there for photosynthesis in leaf is mesophyll tissue, right? The ground tissue of leaf is mesophyll tissue, right? If this mesophyll tissue may this mesophyll which is called palisade mesophyll, can you see it's having elongated type of cells with more numerous chloroplast, right? That's why one side of the leaf is more dark, this side is lighter in color, right? So this is, it will develop more chlorophyll. It is having differentiation in mesophyll palisade towards the dorsal side, and toward the ventral side there is spongy mesophyll which is having less chlorophyll, right? Because this side receives less amount of sun. Now both the sides are covered by cuticle and all, for checking the transpiration rate. Another thing you have to remember that here uh, the number of stomata distribution is also different. The more numerous stomata are distributed toward the lower side or ventral side or abaxial side. Very less number of stomata are present towards the upper side, right? So you have to remember this. You can find certain air cavities also towards the spongy mesophyll side. This is a substomatal cavity. Below the stomatal aperture, you can see some small space is there where the vapors will get deposited so that transpiration can easily occur. You can see here the vascular bundle is also present. Vascular bundle ka size in dicot leaf will not be same, right? If I cut the leaf from this, let's suppose this is the leaf, this will be the thickest one, this will be little thinner, right? If I have it like this, it will be more thin. So if I cut it like this, right, vascular bundle ka size is not same. As we are at the midrib area, vascular bundle will be very big, and we are moving to vein, vascular bundle size will be decreased, right? So what points you have to remember for dicot leaf? You have to remember that this is a leaf which is receiving different quantity of sunlight on both the sides of the leaf. So it's having two characteristic sides, dorsal and ventral. Dorsal side is having palisade mesophyll, more chlorophyll, right? Ventral side will be having spongy mesophyll. There is a differentiation in the mesophyll tissue in the leaf, right, which is having less amount of chlorophyll. It is because more sunlight is there on the upper side, so it will be having transpiration problem. So what should we do? We should distribute very less number of stomata or negligible stomata on the upper side, but on the abaxial side number of stomata will be more, right? And when we'll cut the section, we'll see the vascular bundles in the leaf are not of same size. Midrib will have a larger vascular bundle as compared to the veins. Isobilateral leaf में on the other hand you have to remember mesophyll is undifferentiated because you are getting same amount of sunlight received at every area, right? So what will be the condition? Mesophyll will be of same type everywhere, it will be undifferentiated. Number of stomata distributed will be equal, if one is here one is here also, right? So stomata distribution also equivalent. And when you cut the this kind of leaf, right, let's suppose this is the leaf, here you will be having so many veins which are running parallel to each other, right? There is a no prominent midrib here, right? So when you will cut here, vascular bundles all will be of same size or not, right? Every vascular bundle will be of same size, right? Xylem and phloem like other, it is also conjoint and close type here. So you have to remember all of these characters. Before that, one more thing I'll tell you. In case of monocot leaf, right, if you consider monocot leaf, they have one extra adaptive structure. For adaptation there is one adaptation, right, for water loss, right? For preventing water loss there is one adaptation. What is that adaptation? They have certain cells which will become colorless, empty and large, right? So the cells will become colorless, empty and large. What are these cells? These cells are, okay, I'll draw it. Okay, maybe I can draw like this only, no issue. Always remember the epidermal cells. कौन से epidermal cells? Which are located towards the adaxial side. So adaxial epidermal cells, they will become colorless, empty and large. Why? So that when the water is there in the plant, right, these cells are located over somewhere here, right? All these cells will get lot of water. When the plant is having water, all the water will be going and collecting in these cells. So the cells will become turgid, right? And the leaf can, when these cells, they are turgid, right, the leaf remains expanded. Understood? Okay, because there is no problem in water, water is enough, right? Enough water is present. See, when water is enough, at that time cell will remain turgid. When the water content for the plant is less, then all the cells, they will start losing water and they will come to back to their normal position, so they will become flaccid, right? And the leaf, they will start rolling towards inside. Okay, it will start rolling towards inside, right? This will reduce the surface area, and as a result the number of stomata will become hidden, and as a result what will happen? As a result the transpiration loss will be checked. So only that you have to remember. Now we have to do certain questions. See, which of the following is not a feature of monocot root? Monocot root, they don't have small pith, right? Monocot root is having big pith, more big pith, right? Casparian strips, okay, I forgot to tell you that the suberin containing endodermis, right, it is present in the form of strips both on tangential as well as radial wall, that is also called Casparian strip. So that is the feature of endodermis. Understood? Then the transfer section of a plant part which showed polyarch, radial, exarch type of xylem, endodermis and pericycle, the plant is identified. What section they are talking about? When they are saying we have polyarch condition, we have radial, iska matlab we are talking about root only. We have exarch xylem, confirm root, right? Endodermis and pericycle is there. So if you have polyarch condition, they are talking about which root? Monocot or dicot? Monocot. Okay, monocot root mein everything is more, right? Which is wrong about dicot stem? Ground tissue having collenchyma, sclerenchymatous pericycle, endodermis star sheath, scattered vascular tissue. So what is not correct for this? We don't have any scattered vascular bundle in dicot stem. We have the vascular bundles arranged in ring. Whereas we have semilunar sclerenchymatous pericycle, we have endodermis which is star sheath, and we have a collenchymatous you can say hypodermis, right? So we can say ground tissue which has hypodermis, it is called enchymatous. Okay, in dicot stem vascular bundles are arranged in a ring. In monocot it is scattered, and they are conjoined and open also, right? Because they both have xylem phloem together. Along with that, cambium is there, right? In the leaf of a sunflower, mesophyll cell lies where, right? So mesophyll, okay, so sunflower means dicot they are talking about, and mesophyll is what? It's a ground tissue of leaf. And I told you, ground tissue is always present in between epidermis and vascular bundle. So it should be present where? Adaxial, okay, it should be present between the adaxial and abaxial epidermis. That is the most appropriate answer which is given here, right? Now, which of the following are true for palisade parenchyma of dicot leaf? So palisade parenchyma of dicot leaf, where it is present? It is adaxially present. Yes, palisade is present toward adaxial side, elongated, arranged vertically to epidermis. All of these things are correct for it, right? In the diagram itself you can see it. Monocot leaves have equal size of vascular bundle except in midrib. This is okay, we can consider this. Dicot leaves show different size of vascular bundle. This is also correct. Midrib is little bit thicker, right? But everything else, let's suppose if we consider a banana leaf also. In banana leaf we will see that midrib maybe vascular bundle is more in size, but in the veins the size will be exactly same, right? So that is correct. Dicot leaf, so everywhere it is different. So statement 1 is correct, statement 2 is also correct, right? What is the answer? 1 and 2 both are correct, right? First option itself. So I think this is the last question, or a few more questions. Bulliform cell pe they have asked a question in 2024. They're responsible for what? Inward curling of leaf in monocot, so that transpiration rate can be reduced by hiding the stomata, right? So that's all. I think today we have revised all the memory based chapters which are there for our exam. So many examples are there. Please remember bryophyte and pteridophyte examples I have not added here. So please check it out and please revise it. Many questions possibility are there that they ask from these exams. Last year they have asked many questions from plant kingdom. This year they can go to biological classification or morphology, right? So prepare very well. Have a good mind, healthy mind, keep yourself healthy, drink a lot of water and prepare very well. Thank you.